Hello, everyone, and welcome to today's The Scientist Symposium. I'm Nathan Nee, Senior Editor for The Scientist, and I will be moderating our discussion. Today, our expert panel will explore the latest research concerning CRISPR and its applications. They will discuss the underlying interactions underpinning CRISPR-Cas biology, how to manipulate and engineer CRISPR-Cas systems for new applications or improving existing applications such as gene editing, and how to leverage these technologies for biotechnological and disease research. Before we begin, I'd like to thank GenScript and BD Biosciences for sponsoring today's webinar. We like our events to be interactive, and so we encourage you to send us your questions or comments at any point, and our panel will address them during the Q&A session that follows each presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsors have provided us with some helpful resources related to today's webinar, and we have posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. If you have minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. The symposium will be archived on the scientist's website, and we will send you the link for the on-demand version via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our first speaker. Benjamin Kleinsteiber is an associate professor in the Center for Genomic Medicine and Department of Pathology at Massachusetts General Hospital and Harvard Medical School. He is the Caden Lambert MGH Research Scholar as well. He's a biochemist by training who now runs a genome editing technology development research laboratory that seeks to optimize new methods to modify genetic sequences. Some of the major goals in the Kleinsteiber lab are to develop safe, effective, and versatile tools that can correct genetic mutations that underlie human disease. I think everything looks great on my end from a technological standpoint. So, Dr. Kleinsteiber, please take it away. The floor is yours.
Okay, great. Nathan, can you hear me fine too?
Yes, I hear you loud and clear.
Okay, great. Well, first of all, thanks so much for the invitation to speak about some of our recent research. It's a real pleasure to share some of these recently published stories. So just quickly, some disclosures that I'm an inventor on patents related to the data and technologies that I'll describe to you today. And I have consulting and SAB engagements with various biotechnology companies and venture capital firms. So, of course, I am deeply grateful to the folks on my research team. I have the real privilege of being able to tell you about all of their exciting work, which I'll tell you all about today. So I'm very fortunate to work with a group of talented and motivated and creative trainees who are really doing some incredible science. So as an outline for what I'll hope to describe to you today, I'll spend the first bit of the talk describing some of our recent efforts to engineer CRISPR-Cas enzymes to make them better genome editing tools. One of the goals in my lab has also been to explore the sort of IND enabling aspects of genome editing tool design and begin to use them in therapeutics. So I'll describe to you one recent story of how some of our recently engineered CRISPR-Cas proteins have been utilized now in human. And then if time permits, I'll talk about some of our goals in the future of genome editing tech dev and what that will look like. So I guess, the theme of the future of CRISPR, I'm going to focus on how we can use engineering to improve the tools and what the utility of these engineered tools are moving forward, both for research and for use in the clinic. So, since I'm the first speaker, a really high-level introduction to genome editing. Um, these really are tools that allow us to hone in on specific bases or regions of a large genome within a cell. And the promise of these tools is to be able to target specific regions or bases and make designer changes to those sequences. And for most human genetic diseases, these are caused by single letter changes, but um, there are also lots of other pathogenic genetic lesions that cause disease. So in this pie chart, you can see that the implication of this slide is just that we need a really deep and diverse toolbox of genome editing enzymes that would allow us, in principle, to correct nearly any type of mutation. And I would say, where we are in the field right now is a very fortunate position because we have lots of amazing tools that allow us to address the vast majority of these types of mutants. So the work that we do in my lab is really focused on those main goals. So how do we think about building genome editing tools in ways that will enable new types of genetic therapies? We have a lot of interest in developing and modifying genome editing technologies. One of our core interests is in utilizing a method called protein engineering to make new versions of CRISPR-Cas enzymes with new properties. And then, sort of, all of that is really focused under the lens of how do we do this in a way that enables the development and application of new genetic therapies. So, again, thinking about the future of CRISPR and what this means, maybe just looking backwards where CRISPR came from, these are enzymes that exist in a bacterial cell that are important in defending those bacteria against invading DNA. And what this means is that these enzymes evolved with specific properties that make them a really good component of an immune system that allow bacteria to defend against invading nucleic acids. And some of the key components of these enzymes are the necessity of a Cas9 protein to read a really short DNA sequence called a PAM. And in the context of the native CRISPR system, this is a really important function because in the host bacteria, in the host genome of the bacteria, there are no PAMs encoded near the CRISPR sequences, but those PAMs are found in the invading DNA. And that's essentially a proofreading mechanism of how the CRISPR-Cas proteins can differentiate self from invading DNA. But when it comes to genome editing, the requirement of these big Cas9 proteins to first land on and bind this sequence called a PAM, what this means is we can only target and edit regions in the genome that encode these PAMs. So in certain applications, we may have these PAMless deserts where we can't actually access and edit those regions of the genome using enzymes encoded in nature, what we call wild type enzymes. So this has been a big motivation for my lab and others in the field to try and engineer new versions of Cas9 and Cas12 and other proteins that can recognize an expanded scope of PAMs and now able to sort of eliminate these PAM deserts to enable more precise genome editing. So there's a couple different ways that one could imagine engineering these proteins. One of which would be directed evolution, where you can use a model system either in a test tube or bacteria or yeast to impose some sort of fitness constraint that gives you an ability to select for new enzymes. So in the context of PAM engineering, what you can do is you can try to evolve against a PAM that's not recognized by the wild type Cas9 protein. A second approach is the use of structure-guided engineering, where we have these amazing 3D blueprints of how CRISPR proteins interact with their nucleic acid targets. And you can form hypotheses about how parts of that protein are important for reading and engaging with those nucleic acids. And then you can make custom amino acid substitutions in the protein to try and change those properties. And then a third area in protein engineering is the use of machine learning. And as far as CRISPR-Cas proteins go, I would say this is pretty underexplored. And one of the promises of machine learning is that we can experimentally sample a relatively small space and utilize machine learning to sort of extrapolate those observations to a much larger universe of potential proteins. So the ability to utilize these first two methods to generate training data for machine learning is really important. So when we think about utilizing these methods for engineering proteins, there's a handful of properties that you can sort of envision would be useful to change in these CRISPR-Cas enzymes. One of which is the requirement for these enzymes to recognize PAM. So I mentioned this a few slides ago. And we and others have undertaken lots of different engineering approaches to build new Cas9 variants that can recognize distinct sequences. Of course, though, beyond the targeting range of these enzymes, there's other properties like their on-target activities, how efficiently they edit a specific region of the genome. And then another property like the safety. So ideally, we want to edit only one or few sites in the genome and try to minimize unwanted off-target editing at other positions across chromosomes, minimize that as much as possible. Beyond these three properties, there's of course lots of other potential properties that one could engineer using these different approaches, all towards the goal of having highly efficient and safe genome editing tools. So 5 years ago now, we engineered one genome editing tool that we called Spry that really obviated this PAM requirement constraint. Again, the issue here was that with the wild type Cas9 protein, you can land your Cas9 protein wherever you find PAMs, but there's big sections of genes that remain inaccessible to that enzyme. So Russell Walton from my group engineered this protein called Spry that had a very dramatically relaxed PAM requirement, which meant that it could access a really expanded scope of PAMs. Subsequent to this, the development of Spry, we and others have shown the utility of an enzyme like Spry to knock out genes or small regulatory elements, to be coupled to enzymes called base editors to more carefully position the edit window of a base editor. And then for many other applications. So Katie Christie from my lab developed a method to digest DNA in a test tube using Spry, essentially replacing restriction enzymes because we have an enzyme that can cut DNA nearly anywhere. And then we and others have explored the use of Spry in preclinical use to determine whether or not we can utilize Spry to access regions of the genome that are important therapeutically in ways that were not possible with wild-type Cas9 before. But one of the secrets, sort of Cas9 engineering in my lab is that our goal actually when we were building Spry was not actually to make a PAM-relaxed enzyme. We were trying to take wild-type Cas9 that normally recognizes NGG and change that to something different. So expanding the universe of accessible PAMs by altering the PAM rather than having one enzyme that can access any PAM. And what we've learned over the years, both through our work and that of many others, is that the simplest evolutionary trajectory, we think, is to relax the PAM requirement rather than to change it to something else. And that's been borne out in the literature where most engineered Cas9 proteins have an expanded ability to access new PAMs, but still retain the ability to recognize the original wild type PAM. And very few enzymes have been described that truly just switch the PAM to a different sequence. Again, this really wasn't our original goal to relax the PAM requirement. We think there's a lot of utility in having enzymes with switched rather than broadened PAM requirements. And the reason for that is this. So with wild-type Cas9, we know that the enzyme is actually generally quite specific as long as you have a reasonably specific guide RNA, meaning that it largely targets the intended sequence and can have rare off-target edits. But as we and others develop these PAM-relaxed enzymes, what we found is that they now have improved access to the remainder of the genome, but a consequence of this is that they can encounter more potential off-target sites. So there's some potential safety constraints in using these enzymes. And our goal all along was to sort of take the best of both worlds here, where we could build lots of enzymes that looked conceptually like wild-type Cas9, where they were highly active, but individually very specific in that they could only access small regions of the genome, But together, if we were able to engineer enough of these enzymes, we'd still have the same comprehensive genome targeting as one would get with these PAM relaxed enzymes. So to do this, this has been a longstanding goal in the lab. We needed to build new ways to engineer and characterize proteins. So Russell from my lab, he developed a method that allowed us to profile the PAM requirements of hundreds or even thousands of Cas9 proteins and do that in parallel in a 96-well format. And this was transformational for us because we didn't need to purify proteins anymore to study their functions. And if we had sufficient high quality biochemical data from these types of assays, we could then envision utilizing methods like machine learning because we'd have high quality, really rich data for each individual enzyme. We could utilize that to train models about how the performance of those enzymes equates to their amino acid sequence. And this was one of the sort of original pie in the sky goals when we started the lab many years ago, that Rachel Silverstein, at the time a rotation student, really gravitated towards and became her thesis project over the last couple of years. So in the next couple of slides, I'll describe to you how we've been able to implement these machine learning models to build orders of magnitude greater numbers of engineered Cas9 proteins. Again, this work was led by Rachel. So as a first step, what we had to do is create the training data sets so that we could train the model. And the first thing that we did to do that was build a library of Cas9 proteins that were varied at 6 different amino acids. The theoretical complexity of this type of library is around 64 million different Cas9 proteins because they're varied at 6 amino acids. We can then take this library of Cas9 proteins, we can express them in bacteria, impose some sort of fitness selection, requiring them to recognize one of 16 different PAMs. And then we can take those enzymes that survive through the selection, sequence their PAM interacting domain to learn what amino acids are at these 6 different positions. We can then profile the PAM requirements of these enzymes using our HD-PAMDA assay, again, creating a huge data set now of information about the relationship between the amino acid sequence in the Cas9 protein and its biological properties and how it recognizes a PAM. So Rachel did this for nearly 1,000 different Cas9 proteins. And here's a large heat map to sort of schematize those results, where each row here is the properties of one Cas9 protein. So I'll direct your attention to the left side of this graph, where you can see that a lot of these enzymes that we've clustered together primarily recognize this one row here in dark blue, and that's NGG. You might know that this, though, is the PAM requirement of wild-type SPCas9. So these enzymes don't really offer a new targeting range or a new capability, but what we've learned is that there are lots of different solutions of Cas9s with mutations in these 6 amino acids to still recognize NGG. The second major phenotype in this large data set was this big group of enzymes kind of in the middle of the slide, where you can see that there's lots of blue across each row, meaning that these enzymes have acquired the ability to recognize new PAMs because they've relaxed their PAM requirement. So this is really similar to the enzymes SPG and SPRY that we built a few years ago, where they can now gain access to new regions of the genome because they've relaxed that PAM. But again, that really was not our original goal. We were trying to find more nuanced and unique PAM profiles like some of these that you can see on the slide. And that's where we thought maybe we could begin to use machine learning to learn what the rules were to make these more specific PAM variant enzymes and begin to expand that to a greater number of proteins. So this is exactly what Rachel did. She took these really rich data sets from our HD-PAMDA assay for nearly 1,000 enzymes. She then inputted that as training data into various machine learning models, and then validated, of course, the performance of the models. And once we had a sufficiently good model, she could then begin to predict new enzymes not seen in the training data sets and try to predict what their properties would be. We can now do this actually in principle for the entire data set of all 64 million enzymes, only a really small fraction of which we'd actually characterized experimentally. So Rachel cloned about 240 of these enzymes that we could predict from the Pamela model. And what I'm showing you here now is a smaller heat map where, again, each row is a different enzyme. And what you can see here is that there's very few enzymes that have a relaxed PAM requirement. The vast majority of these enzymes now have more sort of customized PAM profiles. On the very right side of the slide, you can see that we can predict enzymes that target NGG and actually do so quite efficiently. And then in the remainder of this graph, you can see some more sort of designer profiles where we can access really specific PAMs in ways that we just weren't able to observe enzymes coming out of the genetic selection systems. So for the sake of time, I'm not showing you the data. Rachel then, with help of others in the lab, went and validated the performance of these machine learning predicted enzymes to knock out genes as nucleases, and also show their usefulness as base editors to make precise single nucleotide changes. Rachel also showed there were really big specificity benefits in using these enzymes compared to PAM-relaxed enzymes because they recognize a much smaller fraction of the genome. They encounter fewer off-target sites. So here's one potential example that Rachel explored with collaborators at the Mass Eye and Ear Institute, where we could think about predicting a more custom CRISPR-Cas protein to target the rhodopsin P23H allele. So this is a mutation that causes a dominant negative form of blindness, and this is a really challenging sequence for genome editing with SPCas9, because the wild type allele that we don't want to edit has an NGG PAM, but the mutant allele has a non-canonical NGTG PAM. So if you were to use wild type Cas9 for this application, we'd have sort of the wrong phenotype, where the wild type enzyme would target the wild type allele, which we don't want to do. So here what we sought to do is utilize Pamela to predict an enzyme that was highly selective for NGTG, but now unable to target NGGG. And we can't do this with PAM-relaxed enzymes because they can equally target both of these alleles. So Rachel could take the Pamela model and begin to predict lots of different enzymes that in principle would allow us to efficiently access NGTG and discriminate against NGGG. So Rachel created this in silico directed evolution pipeline that allows you to sample all 64 million different enzymes in silico. The results of this specific experiment I'm showing you here just for visualization. Some of the input parameters that you would ask Pamela to utilize would be to maximize efficiency on the sequence you want to edit, like NGTG, and minimize editing on the wild type allele, like NGGG. So here is our starting sequence, which is wild type SPCas9, and you can see it's sort of backwards to what we want. Wild type can effectively target the wild type allele, which we don't want, but is unable to edit the mutant allele. So over rounds of the in silico directed evolution, you can see that this pipeline can basically sample progressively different sequence spaces, eventually enriching for higher and higher efficiency on the intended sequence, the NGTG, while maintaining really low editing against the NGGG sequence. Rachel also then did a similar evolutionary trajectory where she minimized editing across all other sequences outside of NGTG as well. And users can now perform similar in silico evolution based on different input parameters that you would install into the model. So what do these enzymes look like in cells and even in mice? So here, just showing you a HEC293 cell model balance of alleles, where we created a cell line that had one copy of the wild type rhodopsin allele and two copies of the mutant P23H allele. If you use wild type SPCas9, we have the sort of unwanted observation where that enzyme just edits the wild type allele. So this is not good from a disease perspective. If we use a PAM-relaxed enzyme like SPG, you see that it can sort of equally edit both alleles as expected. But again, this is not useful from a therapeutic standpoint. But now using one of a few different Pamela enzymes that Rachel tried, what we observed was really effective editing now of the mutant allele with virtually no editing of the wild type allele. So we've been able, utilizing Pamela, to essentially reverse the biology of Cas9 switching it now to an NGTG targeting enzyme and giving it the ability to discriminate against NGTG. So that's a proof of concept. We obviously would love for folks in the community to utilize these enzymes. So Rachel worked with Justin and Luca Pinello's lab at MGH to design a web tool that allows users to input their specific design constraints into the model and then explore different sequences that come out. So they created a website, and you can basically input your specific starting sequence. Here's the amino acids of wild type SPCas9. You can choose what types of optimizations you want to perform in terms of efficiencies and selectivities and against which PAMs. And then you can run as many rounds of evolution as you'd like to try and identify and enrich for enzymes that maximize your property of interest. Many of these enzymes that have now been prioritized for highest efficiency and selectivity are now available on Adgene. So lots of plasmids already available to utilize amongst the community, but many, many additional enzymes that can be designed using this website. So to summarize the sort of outcomes of this PAMELA study, at a really high level, we think this is a really nice demonstration of the synergy of these scalable pipelines to characterize lots of different CRISPR proteins, and then utilizing those data sets to train machine learning models to predict the vast remainder of the universe of proteins that we just can't sample experimentally. We've learned a lot about the plasticity of PAM interaction with Cas9 proteins and what are sort of some of the rules that govern how Cas9 can access different sets of PAMs. And then the end result from a technology standpoint is a lot of new Cas9 proteins that are safe and efficient and can effectively access regions of the genome that were only accessible before using PAM-relaxed proteins. Okay, so now thinking more, like, again, the theme of the webinar, the future of CRISPR. So we're sort of living in that now. But this started back in September of last year when Kiran Mustanur sent me this email. And essentially he was seeing a patient, a really young patient that was born with a genetic mutation causing a urea cycle disorder. And he found in his lab in preliminary experiments that they could really effectively use a PAM-relaxed enzyme to correct this mutation. But before they sort of embarked on all the effort to begin to manufacture this drug to try and treat this child, they wondered if there was a more specific enzyme that one could use to treat this exact sequence. And I think one of the reasons that Kiran reached out to us is that Rachel had presented this story at Cold Spring Harbor a little over a month before, so he had been familiar with the work that was happening in our group. So Kiran sent us this data in September. Here he basically his lab had just tested SPG and SPRY across lots of different guide RNAs, all of which are PAM-relaxed enzymes, but found a few combinations that were really effective at correcting the pathogenic mutation. So this was great news that they could achieve correction above some level of therapeutic threshold that they thought would be beneficial to this patient. But of course, again, coming back to Kiran's question, could we design a safer enzyme that would potentially have fewer off-target edits and maybe even a benefit in terms of higher efficiency? So we quickly looked at this genetic locus in the CPS1 gene, looked at all the possible different guide RNAs and sort of concluded the same thing that Kiran had, that there was one guide RNA that may be the most effective given the position of the target base to correct this mutation. So we looked in our toolbox for enzymes that could access this NGG PAM. Because in the sequence here, I've annotated all the different PAMs. You can see that none of these target sites have NGG, which means we can't use wild type SPCas9 to make this therapeutic edit. So we can't use wild type Cas9. Kiran's lab found that the PAM-relaxed enzymes were quite effective, but they were wondering, could we use some of these new, more bespoke enzymes to more safely access this region of the genome? So we took two approaches to try and design some enzymes for Kiran's group. First, of course, was to utilize PAMELA to predict enzymes that were really effective at targeting NGCC and doing so highly specifically. And then we also shared some other enzymes with Kiran's lab from another ongoing project in the lab utilizing directed evolution to create more efficient PAM variant enzymes. And this was a more manual project where we were forming hypotheses from the crystal structures of Cas9 to change their PAM requirements and also make them more active. So we shared a handful of enzymes with Kieran's lab, and a few days later, really in the really early hours of the morning, we got this email back from Kieran saying, he hasn't graphed the results yet because he hasn't slept. But he did a quick analysis and found that these 3 new enzymes that we shared with his lab all had much better editing efficiencies than the PAM relaxed SPG enzyme that they had used before. So of course, this was really exciting to us and to him because they had not only potentially safe for editing because these enzymes have a more constrained PAM requirement, but also they were much more efficient at creating the on-target edit. So now I'm showing you some of the work that's published in the paper that was recently described in the New England Journal, where here the SPG enzyme was editing at sufficient levels in theory to be beneficial to the patient. And you could see here, what's called A1 is one of the Pamela designed enzymes that they tried. And then these 2 other enzymes, numbers 2 and 3, were some of the more manually designed enzymes that we had provided to them. Kieran's lab then combined these mutations in Cas9 with a mutation in the deaminase domain of the base editor that was utilized to make this correction. And they found that the Pamela designed enzyme actually, when you added the V106W mutation to TAD-A, retained the highest levels of intended editing. So I think this was a really interesting result because this showed that the machine learning designed enzymes actually outperformed some of the manually engineered enzymes that we've been working on for years in the lab. And these Pamela designed enzymes, you can now at this point sort of just click a button to design. So, of course, there's a lot of really exciting details in this manuscript described by the Musunuru and Aaron's Nicholas labs at UPenn. We played a relatively small role in this to design these Cas9 proteins, but this has been a goal of ours over the last 7 years or so to design these tools for exactly these types of situations where they can be beneficial to patients. So I really encourage you to check out this manuscript. It has a lot of great detail in the IND enabling studies and what it took to really rapidly build this genetic medicine to treat this young boy. And I'm sure you've probably seen lots of news about this. I think some really exciting news that this young boy was able to finally go home for the first time with his family, given that his treatment so far has been effective enough to reduce the burden of his metabolic disease. So a really beautiful demonstration of how these collaborations can be developed to create these custom medicines that can have a tremendous benefit to patients. So to summarize that, I guess I would say that for the future of genetic medicines with CRISPR, I think it still remains difficult to supplant the efficiency and safety even of wild-type SPCas9. It will remain effective at editing sequences where there are PAMs in the right spots. We and others have found that these more generalist enzymes are really useful for research applications and can be quite useful also in therapeutic studies. But moving forward, we're really excited about the use of these more bespoke enzymes to safely and effectively correct mutations in ways that can be maximally safe for patients. And as a framework in how we think about this moving forward, at a really high level, a lot of this work was done at UPenn to identify KJ's mutation. We now are working through next steps in the evolution of the Pamela project to try and make this pipeline as predictable as possible so that users could essentially access the toolbox of enzymes in ways that are as simple as possible to identify safe and effective CRISPR proteins. And our hope is that this will allow us to more rapidly deploy these bespoke enzymes for custom therapies. Okay, so I think I'm almost out of time. So I'll just maybe share 2 slides on what, some forward-looking thoughts on the genome editing field. And sorry as I sort of skipped through some other really exciting in the lab, but I just ran out of time to talk about. So there's been some news over the last 6 months, right, about where we are in the genome editing field and what is really the temperature and health of the genome editing field and where do we go from here. So I think this webinar is really prescient in that sense. And I think behind the scenes, there's a lot of really incredible things happening. We're learning that these medicines are safe and effective in human, and really, as a field in academia and in industry, we're beginning to solve a lot of these really big problems that have prohibited the scalability of these approaches. So, what we've learned actually is that the ability to design these sort of bespoke Cas9 proteins and guide RNAs for each individual mutation, this can be done. We can now do this. This still does take a relatively high amount of time and financial resources and effort. So, there are some bottlenecks in how we think about scaling this process. But I will just say that we're optimistic about how we could solve some of these problems by um, utilizing some of these ML designed Cas9 proteins to build a sufficiently large toolbox of enzymes that people could essentially take off the shelf and implement as known quantities into new genetic therapies. And as a second approach, where I think there's a lot of interest in the community, is developing mutation agnostic editing approaches that would obviate the need to correct each individual mutation. One of which are these larger scale editing approaches that can be utilized to either correct exons at a time or even insert large corrective cDNAs at programmable regions of the genome. And by doing that, one might be able to develop a few or maybe even only one genetic treatment for all patients that have different mutations across a gene. And as we run quickly towards that promise in the field, I think it's a really exciting time to solve some of the big challenges that lie ahead to begin to realize the promise of these types of editing approaches. So, to summarize, I think obviously we've learned a lot about how CRISPR proteins function and begun to really uncover the potential of these naturally occurring tools as genetic therapies through the use of protein engineering. This has allowed us to create a really versatile, safe, and efficient toolbox of enzymes that give us the flexibility to correct genetic mutations in ways not possible before. So, with that, I'm again really happy to acknowledge all of the folks in the lab. I'm realizing I need to update this slide because we've not got some new folks in the lab this summer. But lots of people contributed to the work that I described to you today. I'm, of course, really deeply grateful for all the funding sources, especially our NIH funding that enabled a lot of the work that I described to you. Thank you for your attention, and I'd be happy to answer any questions.
Thank you very much for that wonderful presentation, Dr. Kleinsteiver. So let's just launch into our Q&A session, and I guess while we wait for some more audience questions to trickle in, you mentioned that Pamela had essentially kind of caught up in terms of performance to some of the enzymes that you had been engineering, I presume over a more prolonged period of time. And I was curious as to your thoughts on that. How exactly has Pamela caught up, and whether or not this is kind of a one-off, something that happens once in a while, or have we kind of reached a situation where machine learning can consistently match the products of kind of prolonged engineering efforts?
Sure. Yeah, I think the ability of Pamela to predict good enzymes is dependent on having good training data. And I don't mean to trivialize the amount of effort that went into building Pamela. That took Rachel 5 years and a lot of efforts in the lab by other folks to build some of the experimental systems to do this work. So I think we're at a really fortunate time now in the field, though, that we can use a model like Pamela to predict new enzymes and to have them perform as effectively as really, state of the art enzymes that existed before or remained unpublished. So I think we hope that this type of performance will extend to other functions in the genome editing toolbox, other types of enzymes like CRISPR associated transposases or maybe even recombinases, where if you can characterize enough of these proteins, you can then train other machine learning models to predict new versions of those tools as well.
Thank you very much. For our next question, how do the emergence of these tools affect how we approach polygenic diseases?
Yeah, so I think that's a really interesting question. So a lot of the genome editing tools that I just described in this seminar, I think they're probably most effective at monogenic diseases because you can focus on just one mutation. But depending on how polygenic the disease is, one might be able to multiplex edit a few different types of corrective mutations into a patient. Or we're really excited about these pan mutation editing approaches where one could either install a corrective DNA sequence into an endogenous locus in a programmable way, or utilize these protective genetic variants that, there are individuals walking around who are essentially immune or protected from different types of polygenic diseases. And that by installing a single letter edit in one gene in that patient, you might be able to reduce or even eliminate the risk for a polygenic genetic disease. So I think genome editing has a lot of potential for polygenic disease, even though at face value it's a little complicated to start. There are lots of opportunities to leverage these technologies for all kinds of disease.
Thank you very much. For our next question, when using these flexible Cas9 proteins, what would the workflow look like in order to prevent editing of the template cDNA?
Yeah, so the guide RNA itself doesn't edit the template cDNA. Oh, I see. The question is for the large sequence insertion tools, how would you prevent self-targeting of your cargo? I think it's relatively straightforward. You can just simply recode some of the amino acids in your cDNA so that once that's inserted into the genome, it's essentially immune to targeting by the Cas9 protein that's been designed to target the native genetic sequence. So that's a good question. Something that people have to be cognizant of when they're designing these insertion approaches to make sure you're not inserting into the cargo that you just inserted into in this sort of perpetual cycle.
Thank you very much. And I think we can wrap up this Q&A session with one last question. And I was curious about what are the next steps for Pamela, improving Pamela, or maybe building new models down the line?
Yeah, I think there's a lot of really exciting implications of the work because what we've learned sort of obviously is that as we add more data to these types of models, they become better and better. So um, I think if I could convince enough people to screen, thousands more enzymes, we could create better and better models that can predict more confidently a larger universe of proteins. And then I think obviously the ability to extend this type of framework to other properties of Cas9 or to other enzymes entirely would add new fundamental capabilities to the genome editing toolbox. So I think there's lots of exciting opportunities moving forward.
Thank you very much for answering those questions, Dr. Kleinsteiber, and for again, a wonderful presentation.
Okay, thanks so much. Really appreciate it.
So with that, let me introduce our next speaker. Mitchell O'Connell is an associate professor of biochemistry and biophysics at the University of Rochester and a member of the Center for RNA Biology. After completing his PhD in biochemistry at the University of Sydney, he joined Jennifer Doudna's lab at UC Berkeley as a postdoctoral fellow, where he made key discoveries about RNA targeting CRISPR-Cas systems. His current research focuses on understanding RNA-mediated gene regulation and the development of new CRISPR-based tools, as well as the behavior of membrane proteins involved in antiphage defense. Okay, let's just make sure everything. Let's just take a look at everything from a technical perspective.
Just gonna put this up. You can.
Wonderful. I can see your screen. Everything looks great, and I hear you loud and clear. So please take it away.
Thank you. Well, thanks so much for the invitation to this symposium, and really excited to talk about some of our work on CRISPR-associated membrane proteins and membrane proteins in general in antiphage defense systems. And so this is going to be quite a different talk from Ben's talk, which was wonderful. It's really cool to see all that engineering. We're really starting to dig down into some of the new biology we're discovering with these systems. So I'm going to take you through how some of these Cas13 containing CRISPR systems have these additional membrane proteins, and we've really, I think, figured out that they act as downstream effectors to really boost antiviral defense. So Ben did a wonderful job of giving us a CRISPR-Cas background, so I will summarize that very quickly. And then I'll very quickly talk about Cas13 as an RNA targeting CRISPR effector protein, just to kind of orient you about its enzymatic activities. Then we'll really dive into one of our studies where we looked at this small membrane protein called CSX28 and how it's really required for robust CRISPR-Cas adaptive immunity. And then I'll talk about some newer unpublished data on understanding how CSX28 is actually communicating with Cas13b. Got some really hot off the press exciting stuff there, and some of these other membrane proteins that are also in CRISPR-Cas systems, such as CSX27, and how they're behaving similarly, and also the role of some of these proteins in Casless defense systems, because we're finding them scattered throughout a lot of these defense islands in various shapes and forms. And so, as many of you might know, CRISPR-Cas immune systems use small RNAs to mount an adaptive mobile genetic immune response, whether it's against viruses or plasmids or other mobile elements. And they use a CRISPR locus that contains spaces that match these viral or mobile genetic element DNA, or in some cases RNA, and a associated, sorry, I'm just going to get my laser pointer so I can see it a bit better. A number of associated proteins that all act to mount this immune response. And the way that this works is you get this the transcription of the CRISPR locus into basically a long non-coding RNA in most cases, known as a pre-CRISPR RNA. This gets processed either by dedicated Cas enzymes or host factors to form mature CRISPR RNAs, which are loaded. That was frustrating. What happened there? Loaded into these RMPs, which are now competent to surveil the cytosol, where they're looking for either base pair complementarity with the guide RNA, the CRISPR RNA, between RNA or DNA. And our lab's really been focused on these RNA targeting enzymes and how they're able to function and work to prevent phage predation. And so the result of this base pair complementarity leads to DNA or RNA degradation. And in order for all this to occur, as we know, there needs to be an upstream integration step that uses a dedicated integrase to integrate pieces of DNA into the CRISPR locus for future surveillance. And so these CRISPR-Cas systems come in many flavors, and this is a slightly older slide, but it really does illustrate the diversity of CRISPR-Cas systems. So we often think of Cas9 as CRISPR, but CRISPR is also many of these other systems, including these large, complicated class 1 systems and the more abbreviated class 2 systems. And our labs really kind of focus most of their time over the last couple of years understanding this enzyme Cas13 and how does it really work to target RNA, which I'll go through in a minute. And more recently, as we're seeing and sequencing more genomes and identifying more of these systems, are there downstream effectors that elicit additional antiviral activity? Our lab's really got interested in this because of this interesting connection between these nucleic acid enzymes that are cleaving RNA in this case and some proteins that reside within the membrane of these organisms, and how are they communicating with each other. It's really opened up some really novel biochemistry that we're excited about. And so to orient you how Cas13 works, this was work that was done during the end of my postdoc, with teaming up with an excellent graduate student in Jennifer's lab at the time. And through this and work of many others in the field, we can kind of put together a model of how this enzyme works. So the enzyme contains these two hep-N domains, which are known nuclease domains. They come together close in 3D space to form this kind of, one might like to think of it as an intramolecular dimer, where the active site is formed in the interface between these two hep-N domains. You get binding of Cas13 to a pre-CRISPR RNA. There's a separate active site within Cas13 that's able to cleave that pre-CRISPR RNA, and that was kind of novel at the time. to generate these mature complexes, binary complexes, where these hepends exist in an inactive state. And then upon target RNA binding, you get this really nice conformational change where the hepends kind of slide across over each other, and you create this active site. And so what was interesting about this active site from biochemical and also structural perspective, it is very solvent exposed, and it's not facing down on the target RNA, guide RNA duplex to cut down in here, as we see with Cas9. And so this kind of, the positioning in this active site kind of leads to this interesting behavior where you have cis target cleavage, possibly, and this is still a little controversial, but what is well known is that you have trans target cleavage where upon activation of this enzyme, if there's sufficient other RNA present around, it can also cleave that. And there is what appears to be in vitro nucleotide specificity, so different orthologs of Cas13 may cleave after different nucleotides, but it's relatively non-specific. More recent data, which I will get into in a moment, suggests that in vivo and under more controlled conditions, these enzymes tend to have at least some of them, it's not clear across the whole universe of Cas13s, but some of them tend to have a specificity for cleaving tRNAs. And this becomes really important when we're thinking about these membrane proteins. And they leave these interesting 5' and 3' cleavage products that become important in terms of the downstream sensing of these cleaved RNAs. And so when I started my lab, I was really thinking about ways to get back to doing some biology. We've done a lot of tool development during my postdoc, and it's a hyper-competitive field. And I just got excited about going back and doing some kind of more discovery biochemistry. And so I had one of my first graduate students was excited to work on this protein known as CSX28. And so all we knew at the time is it's, as a monomer, it's approximately 180 amino acid protein. It had this predicted transmembrane region. There was some controversy about whether it was actually a transmembrane protein, but I hope to show you that it definitely is. And there was some evidence in the literature that it was shown to enhance Cas13-mediated antiphage defense. But the kind of mechanism by which this was occurring was really not well understood at the time. And so for this project, as I said, it was my first graduate student, Erica Vanderwall, was very brave on taking on this protein, as we weren't a membrane protein lab at the time, but we're slowly becoming one. And a staff scientist in the lab, Bogdan, and we teamed up with structural biologist Liz Kellogg and her really wonderful graduate student, Jung-An Park, to kind of structurally characterize these proteins. So the specific questions that we had were, what are the requirements for this assay, for this activity? And we can use a battery of phage defense assays to really get at what was happening when this membrane protein was activated, and really how that was happening biochemically and structurally. So we used a combination of cryo-UM and a bunch of in vivo membrane function assays to really tease apart what was happening with this protein. And so one of the main assays we use, just to orient you about some kind of classic phage defense assays that are used in the field, are these efficiency of plating assays, or some folks like to call them efficiency of plaquing. And they can really give you an idea of, for a certain host system, their susceptibility to phage predation, phage infection. So the way we do this, we did it for these experiments, is we had what we call a 2 plasmid system where we have Cas13b, a CRISPR RNA on one plasmid that is designed to target a phage of interest. In this case, we're using lambda phage, but we've spread out since then to look at a whole bunch of different phages. And we have on a second vector that's compatible, CSX28, and the corresponding non-targeting or empty vector controls. And so what we do, we can transform lab strain E. coli, MG1655s, for example, with these plasmids and generate a soft agar double overlay setup where you basically end up with a lawn of bacteria embedded in agar. And then you can literally just take a dilution series of phage and spot it across that agar, and you will, if you don't have any protection, you'll see a lot of zones of lysis, so bacterial cell death, where these phages are propagating and then bursting the bacteria. Or if you have really robust defense, you will only see these kind of these zones of lysis at higher concentrations of phage. And so we can use this to really understand some of the parameters of the defense mechanism. And so you can quantify this as an efficient plating. So you can set an untransformed wild type E. coli strain at 0, and the axis goes down. And so the lower the axis, the numbers on the axis, the decreased susceptibility to phage. So a decrease in efficiency of infection. And so we can do this with either Cas13 alone with a guide of choice that we can design against lambda phage transcripts, or we can do it in the presence of CSX28. And so in the absence of Cas13 or a guide RNA, we really don't see any defense. It starts to get interesting once you add a guide RNA. And for this one, guide RNA 1, this Cas13 is a pretty weak defender of lambda phage, but you're getting about a 1.5 log-fold improvement in defense. And what was really exciting for us is once you add CSX28 to the mix, you get this massive boost in defense. So now it's almost to the limit of detection of this assay, which is around 10 to the minus 5. And so really just protecting this system against phage. You can do this across a range of different CRISPR RNAs that target different transcripts, and you see various effects depending on the strength of that guide RNA, CRISPR RNA, but we do see really large increases in defense. And one of the first interesting insights that we made was that you can make a single point mutation in one of those hep-end domains, a single histidine to alanine mutation, and you completely lose the ability to communicate to CSX28 to get that boost in defense, giving some of the first hints that enzymatic activity of Cas13 is possibly required for the activation of this increased defense by CSX28. And so then you can look at the other side of the equation and see how the host grows upon activation of this system to see if it's a dormancy, bacteriostatic, or cell death kind of phenotype that's responsible for the kind of protection against phage, a term known as abortive infection. And so what we can do here is we can monitor the growth of bacteria over the course of an infection at time 0. And for an untransformed cell with an infection of 0.2 MOI, these cells grow, and then the population eventually crashes and lyses. And that's how you see this population crash, decrease in the optical density. Cas13 alone helps delay this lysis a little bit, but it still is overcome by the phage infection. And it's really not until you add CSX28 alone, CSX28 in combination with targeting Cas13 that you see this kind of bacteriostatic abortive infection type phenotype. They're growing, but they're much slower, but they're beating the phage infection. And so without going into too many details, we were able to purify this protein, and to our amazement, it came off. We purified it in a detergent, DDM, and it was coming off as optimers on size exclusion and laser light scattering. And so we got excited about the potential for cryo-UM, given that it was now of a size that could be possibly solved. And long story short, some excellent work by Jung Ang and Elizabeth was able to take our purified protein, put it on graphene oxide grids, which enabled us to collect a sufficient amount of data to get a 3.7 angstrom cryo-UM model that forms this octameric structure with a pore-like center that's embedded within the DDM mice cell. And so the N-terminal TM domains are embedded within that detergent. You can take this map from the cryo-UM reconstruction and build a model in very easily. Side chains are pretty easy to see in this model, at least in the cytosolic portion. And that forms this kind of 4 alpha helical bundle kind of fold with that N-terminal helix going up into the detergent mice cell, suggesting it is probably membrane localized in vivo. This pore size is around 5 to 10 angstroms, depending on how you measure it. So it's large enough to be relatively non-specific in terms of ion permeation, small ion permeation, but probably not large enough to let things like ATP through. But we're now collaborating with folks to actually measure the ion permeation properties of this enzyme now that we have the ligand in hand. And that's pretty exciting for us. So once we saw that it was a pore, we really wanted to try and see what kind of it was doing to the membrane during infection. And so one way to do this is we had this hypothesis that maybe it was opening up after some kind of interaction with Cas13 or some kind of ligand binding. And we have data that I'm not going to show today that it's unequivocally sitting in the inner membrane of E. coli when we express it heterologously. And we could do an experiment where we can measure membrane depolarization upon activation. So you can take this dye, which is a voltage-sensitive dye known as dieback, that is weakly fluorescent when there's a proton motive force or a membrane potential across the inner membrane. You can think of the inner membrane of E. coli much like the membrane of the mitochondria where the electron transport chain sits and the ATP synthase. So this potential is really important to drive ATP production. And so if there is pore formation or opening, the fluorescent dye becomes strongly fluorescent, and you can measure that using either a plate reader or, in our case, we use flow cytometry because we get a lot more richer data set, and you can assume that there is a loss of membrane potential with this dye. And so we did that experiment, and long story short, we see really nice membrane depolarization caused by CSX28. And so in the experiment, this is a flow cytometry experiment with that dye. As a positive control, we can use polymyxin B, which is effectively an antibiotic. It's in some of those skin products that you use to treat cuts and wounds. And it punches holes in gram-negative proteins. And so you see this strong depolarization. And then you can set up an experiment with lambda phage infection. At 0 minutes, they're all nicely lined up and you're not seeing any depolarization. Upon lambda infection, you start to see what's probably most prominent is this group when you have Cas13b and CSX28, this highly depolarized group of a population of cells within the culture. And this grows over the course of the cycles of infection. And this can be quantified and really matches the kind of MOIs we're using in these experiments, seeing up to 40% of the population depolarized by 90 minutes. And so this was pretty exciting for us because it kind of fit with our hypothesis that this pore-forming unit is probably modulating metabolism in some way. And so the next experiment we looked at is a very kind of crude experiment to look at the kind of metabolic state of the cell. And you can use a dye known as resazurin. It's basically a viability dye. And it basically reads out the NAD plus status of the cell. And so there are reductases within the cell that are able to reduce this and create a really high fluorescence pink product. And those reductases require NAD as a cofactor. And so we can do this experiment during phage infection. And what we see in this is that in untransformed cells, you get this really rapid turnover of resazurin into its product. And that flatlines as you've used up all the substrate. We have this weird dip that's a funny quantum mechanical phenomenon known as a dark state switching, but I wouldn't read into that too much. The take home here is that when you have CSX28 and Cas13 in the presence of a phage infection, you get this really slow turnover of this resazurin dye. And it takes almost 3 times as long for that population of cells to turn over resazurin as cells not expressing and activated CSX28 in the context of a Cas13b cleavage event. And so the take home from this is that the opening of the pore is leading to lower cellular respiration and overall cellular metabolism. And you're basically turning off the lights of the factory. And that, as the phage require host metabolism and host building materials, you stunt viral replication and infection. And in data I'll show you in a little bit, that's part of another project. And I didn't have time to show it today, is that we now have the ability to do ATPase assays in the lab, or ATP production assays rather, and we notice that we get a large crash in ATP levels upon the activation of this membrane pore. And so this all leads us to a model of how this is working. And before Matt in the lab started, it's an excellent graduate student in the lab, we had this model where we really weren't sure, but had some hints about how the activation of Cas13 and its cleavage of RNA were able to activate this membrane depolarization caused by an active CSX28. And so at the time, we had really no evidence that these can form a co-complex. We tried many things, including co-IPs or co-complex formation via size exclusion. And that one hint that I mentioned before about making a single point mutation in the hepban active site was unable to activate CSX28. And so we kind of turned to a second model where we think it might be actually the RNA cleavage products that are interacting with this protein. And we have a lot more evidence for that. And I'm going to show you the really clear evidence that we now have from some RNA-seq experiments. And so we know it combines specific RNAs in vitro and in vivo from ENSAs, pull-downs, and cross-linking experiments. We know in vitro with purified components, we can cross-link RNA to form covalent oligomers, which kind of hints at where this RNA might be binding on this protein. And we've collaborated with Alessio Accardi and have some really nice preliminary liposome flux assays that show a dose-dependent increase in chloride flux when using an imperfect RNA ligand. We're now repeating those experiments with what we think is the correct one. And so we're pretty excited about this new kind of phase of understanding how these membrane proteins are actually RNA-gated, potentially RNA-gated channels to modulate this immunity phenotype. And so one big hint that we got that we might be able to track down what this ligand was, we had these ideas that maybe it was binding non-specific RNA that was cleaved, but then this really nice paper came out of Ekaterina Semenova's lab, where for Cas13a at least, they were able to do an experiment where they could map cleavage products over real time under very specific conditions in E. coli. And so this was using a much more careful approach with making sure you were inducing at lower levels, that you were really catching early time points. And they were unequivocally able to show, at least for LSH Cas13a, that tRNAs were popping up as the main cleavage products very early on in the reaction. And one could say that's because they're hyperabundant, and that's probably true, but it wasn't all tRNAs. It was a very specific subset. And they were able to go on and use primer extension assays to really map experimentally where these cleavage sites are. And the take home was that it seemed to be occurring in the anticodon loop with a preference for wobble-based uridines. And so this is for one specific Cas13a ortholog. It doesn't have CSX28 next to it. And so we went on and collaborated with Ekaterina and repeated these experiments for several other orthologs of Cas13. And here we're particularly interested in PBU Cas13b because that was the CSX28 Cas13 that we were using. And we see a very specific subset of tRNAs being cleaved by this enzyme in the same kind of setting. And so we thought, let's do an experiment and see if we can do pull downs with cleaved Cas13 small RNA products, given that we had this hint, and just see if they bind CSX28 in an in vitro pull down. So we can purify CSX28. We can purify uncleaved and Cas13b small RNA products, either from a phage infection, but we also have now plasmid targeting systems that we can induce to activate Cas13 and also CSX28. And then simply an old school mix and incubate, add nickel or in our case, we are actually using cobalt beads. They work a little better. And then we can wash and elute either using TEV protease or just strip it with a phenylchloroform extraction. And these are the kind of RNAs that we can purify from this kind of experiment where you can use a specific protocol to purify the small RNA fraction, given we really wanted to focus on those tRNAs, and take a couple of different orthologs, and you see that you can see the cleaved RNA products being formed and being able to be purified. And so then we can basically go ahead and do that pull down and just see what happens. And to our surprise, we saw something pretty striking. And so you have these really kind of heterogeneous inputs where a lot of things are getting cleaved by Cas13. You do the pull down, you do several washes, and what we really saw was two really distinct bands that correspond to what we think are nicked tRNAs. And this occurred across several different orthologs of CSX28. And so we got really excited about this and wanted to figure out what their identity is. And so we did a very similar RNA-seq experiment, this time in collaboration with MesoRNA that have a platform technology to really pull out tRNA sequencing. tRNA sequencing is inherently difficult because of all the modifications in the tRNA that make it hard to make libraries. And so they have some techniques to get around that. And to our excitement, we saw that we had really one hit that was hyper-enriched relative to all the other tRNAs that were depleted, and this second smaller enriched hit. But we got really excited that there was one really standing out. And so here we're looking at a volcano plot for the differential enrichment of tRNA abundance, I should have mentioned, from a pull down versus that really heterogeneous input sample. And so it's quite striking that we're really seeing one product pop out. And what's exciting about this product here is it's also the same tRNA that was ID'd in the pull down. It was the same RNA, I think I don't like how I put this, but it's the same RNA that was the highest enriched cleavage product that was identified in Ekaterina's RNA cleavage experiments. So the product that gets cleaved the most is also the one that seems to be binding with the highest affinity and specificity for CSX28. And we're not quite ready to share the identity of that product, but we're doing a lot of biochemistry now to follow up and make sure that we really do have what we think is the correct ligand for Cas13 that is binding CSX28. And so that story is ongoing, and we're pretty excited to finally kind of figure out how that ligand is gating that ion channel membrane pore. And we've kind of turned our attention to many of the other membrane proteins that we're seeing coexist in these operonic structures with Cas13. And so now there's a lot of work in the lab working on another membrane protein that bears no resemblance to CSX27, to CSX28 rather. It's a completely different protein. And this one instead contains 4 transmembrane domains within a single polypeptide. Previous work on it had shown that it can inhibit Cas13 mediated antiphage defense, but that was in their experimental setup, and turns out there were some cloning issues in those experiments that have kind of muddied the results. And so we went back and took a fresh look at this, and we're excited to be able to say that it pretty much acts in a very similar manner as CSX28, that it acts to boost defense by Cas13b. So we see that same bacteriostatic phenotype where upon activating CSX27, you get this slow growth phenotype, but in its absence, it's not able to protect against lambdaphage in this case. We also see this really nice depolarization upon activation, and in a very similar manner to CSX28. And of interest, now that we have these ATP set up, we see this massive drop in ATP activity upon activation of this enzyme. This is also a experiment that I was alluding to before with CSX28. We see the same phenotype, so this depletion in ATP. And something that we've found interesting is a lot of the Cas13s within these CSX27 containing systems are relatively robust. And so what we find is that we can start playing around with the idea of phage mutational escape. And we show that when you have mismatches within the guide RNA, you can really dampen the ability for Cas13 alone to help, when normally it would help pretty robustly. But CSX28 is able to come in and save the day and help as a second line of defense to really prevent phage mutational escape, because it can really prevent that population from exploding. And with the kind of advent, and this is just my last couple of slides, with the advent of AlphaFold and structure homology searches, we've really been able to build a pipeline to look for these proteins all across the kind of prokaryotic space. And what we're finding is that they actually exist in many other operonic systems that don't contain Cas13s. So we even find them in E. coli. They look quite similar at the structural level. And you can do these kind of defense island guilt by association approaches to really look for the genomic locations of these and see what systems they're lying next to. And so we're seeing other nucleases that have been pretty understudied sitting next to CSX28. We're seeing reverse transcriptases, these chat domain containing proteins that form these kind of ancient caspase-like systems. And just a really whole universe that we're excited to start exploring. It's almost a bit overwhelming. And so to finish, hopefully I was able to convince you that there's this whole world of membrane proteins that are sitting within CRISPR or other phage defense systems. And we're really trying to think, at least the ones that we've worked on so far seem to be gated by nucleic acids, which is quite unprecedented in terms of at least transcribed nucleic acids. And so to finish, I'd love to thank everyone in the lab that's able to help with all these projects. It's just really wonderful to work with everyone every day, all the people that have been in the lab previously, our funding sources and our collaborators, and I'm happy to take any questions.
Thank you very much, Dr. O'Connell. So let's get into the Q&A session here. And I might have missed this, but can I ask how Cas13 signals CSX28? Is it by cutting, signaling, or a different mechanism?
Yeah, sorry if I wasn't clear about that. And so the mechanism is that we think it's able to, Cas13 is actually specific for tRNAs in a way that we didn't appreciate before. So it's cleaving in the anticodon loop of a specific tRNA that we've discovered through that RNA-seq. And then that tRNA is now a nicked tRNA that looks different from other degradation products in the cell. And we're finding that nicked tRNA has an incredibly high specificity for CSX28. And we hypothesize that the binding of that nicked tRNA, which we've now demonstrated, we have really beautiful size exclusion traces showing that interaction, is able to gate the channel and open it so it can let ions through. So it's really a nicked tRNA that's actually acting as a ligand, which is really wacky, and we didn't expect this to be the case, but there is some precedent for nicked tRNAs acting as signaling molecules. And there are actually phage enzymes within, say, T4, for example, that actually repair them to get around this as an anti-phage defense.
And how specific is this mechanism? Are there other nicked tRNAs, for example, that do other things that are also mediated by Cas13 or?
Oh, yeah, that's a good question. So yeah, cleaving in the anticodon loop of tRNAs there is a lot of precedent in toxin antitoxin systems and various nucleases, both in prokaryotes and eukaryotes. I think what you're getting at is we know that Cas13 is nicking other tRNAs, and maybe they're branching off and signaling to some other host proteins that we haven't yet discovered. We do think about that but at least for this two-component system, we think it's really one tRNA that's doing all the damage. The other tRNAs being cleaved might also help with translational repression as well. We really see quite a diversity across the Cas13 space of which tRNAs are getting cleaved, how many of the whole tRNA pool is getting cleaved, and then there are even some enzymes that are very generalist Cas13s that will cleave a lot more than just tRNA. And that we think that mechanism has kind of diverged from these more specific tRNA cleaving Cas13s, if that makes sense.
Thank you very much. How prevalent is this kind of signaling activity as opposed to gene editing activity across different Cas proteins?
Oh yeah, that's a great question. So we haven't looked into Cas9 and kind of the death by a single genome cut kind of approach. But if you look in type 3 systems, for example, and I think there's even evidence of type 1 class systems too, but type 3 systems have a pretty sophisticated signaling mechanism where, so these CMR or CSM complexes actually generate can generate oligoadenylates that are cyclic, so multiple adenylate molecules joined in a circle, and those can then go on and bind to things such as CAF domains that can either turn on other heparan nucleases as kind of a second line of defense, but there are also CAF domains that are associated with membrane proteins that form membrane pores. And some of Malcolm White's work has done some really nice stuff on that. And we think it's probably just scratching the surface. We're seeing these membrane proteins kind of, as Eugene Koonin would say, guns for hire, that are kind of being dropped in and out of these defense systems at a pretty rapid rate. So we're finding them associated with a lot of other enzymes that aren't necessarily CRISPR enzymes.
All right, that's about the time that we have for this Q&A session. So I want to thank you again, Dr. O'Connell, for your answers and for being here with us today.
Thank you.
As a reminder to our audience, before we move into our next speaker, you can submit your questions at any point during this webinar, and we'll get to as many as possible during our Q&A sessions. Next, we have Dr. Patel, who is the Senior Segment Marketing Manager for Cell Therapy at GenScript USA Incorporated. She focuses on delivering comprehensive solutions for cell and gene therapy development and leads strategic marketing initiatives and market research in this field. Before transitioning into marketing, she served as Senior Scientist and Technical Product Manager at GenScript, contributing to the development and commercialization of innovative products. With over a decade of experience in both academic and industry settings, she brings a strong scientific and strategic perspective. So, Dr. Patel, please take it away.
Hi, everyone. Thanks for joining us today. And I'm really excited to walk you through how GenScript is supporting CRISPR-based gene editing program, starting from early stage research to all the way through the clinical trial. So here is the quick look at what we will be covering today. We will start with the current cell and gene therapy landscape and also include what's new happening in this area. And next, I will introduce GenScript CRISPR toolbox and shows how our reagent and service support every step of researcher journey. And at the end, I will share a few case studies. So let's start with the cell and gene therapy field. It's like growing quickly, and scientists are constantly driving the new discovery and then reshaping how we approach the new treatment. So let's start what's happening in the global level. So in the CGT industry, like until 2024 globally, for clinical use, more than 100 cell gene therapy has been approved, and which include like 72 non-genetically modified cell therapy and 33 gene therapy, which include the genetically modified cell therapy also. And interestingly, there are over 4,000 therapies are currently in development, and all of these, like out of all these, 2,000 are gene therapy, which include the genetically modified cell therapy also, and others are other remainings are like non-genetically modified cell therapy. And when it's come to genetically modified cell therapy, CAR T cell therapy remain the most common technology in the pipeline, which accounting for 49%. And as that the global cell and gene therapy landscape expanding, it's clear that the cell and gene therapy are truly reshaping the personalized medicine. And while cell and gene therapy use different approach, both often rely on gene editing to make precise changes to the genome. And scientists have made a long way from early tools like Zephan and Talon to the innovative CRISPR-Cas9 system. But the progress didn't stop there. They keep bringing the newer technique like base editing, prime editing, which is now often now offer even more safe and more accurate editing technology. And as part of this ongoing approach, we see like in 2023, we saw the major milestone, the first approval of Casgevy, which is a CRISPR-based therapy for sickle cell disease, which modified the patient's stem cell to increase the production of hemoglobin, which helped to prevent the sickling of the red blood cell. And that's like mark as beginning of new chapter in precise gene editing. And another example is like NK510, which is also received IND approval into the last December. It is kind of universal NK cell therapy developed by Base Therapeutic using the CRISPR-based cytosine-based editor, where and so this technology used to convert cytosine to the thymine with nearly zero off-target effect, and it's considering as accurate and efficient genomic modification. So together, this example shows how the next gene editing tools are translating into the real therapy, and it completely opened up like new possibility for safe and more effective treatment. And now let's quickly review how the key CRISPR system used in cell and gene therapy. So first is the traditional CRISPR-Cas9 system, which is the most common. It cut the DNA to either knock out or fix the gene, and widely used in the CAR-T therapy. And the next one we have is like the Cas12 or Cas no, which is also a kind of CRISPR cell therapy, which cut the DNA differently and creating the sticky ends, so which help to make more precise edit, and it's great for complex gene change. Another one is the prime editing. So it's a newer method that fixes the mutation without the cutting both the DNA. So it's very useful for kind of treating the disease with precision. And another one is the base editing, which also just changed the single letter in DNA, ideal for point mutation. And RNA editing also getting more attention. So that's what it's doing. It's not cutting the DNA, but it edits the RNA to adjust the protein level. And it's quite useful in neurological disorder. So finally, CRISPR activation and interference also used to turn the gene on or off temporarily, and this help in immunotherapy or controlling gene related to tumor growth or inflammation. And each system use specific enzyme, guide RNA. Some also use the template or special reagent to help with this editing process. And no matter the modality or the editing relies on the right combination of enzyme, guide RNA template, and the delivery system. So, and GenScript is exactly specialized in this area. So at GenScript, we provide all the necessary reagent with the phase appropriate form. And this means you can dream whatever want to do in your project, but we are here to help by providing different kind of tools and technology to support your work. So let's start with the heart of the every CRISPRs is like Cas9 protein, and which is the core material to carry out the gene editing. And as we know, the quality of nucleus has directly impact on the success of gene editing. So at GenScript, we offer like different kind of Cas nucleus, and we have license from the Broad Institute as well as the David Liu Lab for the base editor and prime editor. And we offer like Cas9, Cas12, even Cas13, and base editor protein, which is the adenine base editor, cytosine base editor, and the prime editor protein. And all these are available into the research format, means ready to use as an off-shelf product. And we also giving from custom service for when you moving from research grade to going from clinical stage. So anyone can confidently move from research through clinical development. And we also provide like formulation and delivery support, so that can help to tailor the CRISPR tools for more efficient and targeted application. So let's go through some slide where reflecting the quality and the efficiency of the Cas9 system. So to confirm the specificity of Cas9 system, the study was carried out by Dr. Feng Lab and in the Broad Institute. And in their comparative study, they reported that they found that GenScript Cas9 shows better specificity, high editing efficiency, and lower off-target activity than commercial hi-fi Cas9. And our Cas9 system, we also validated in-house, and comparative study shows it has lower off-target effect compared to the other vendor, and while maintaining similar off-target effect compared to other vendor, while maintaining similar on-targeted editing efficiency. And let me share more some data which from the test we did using human primary T cell to check out the Cas9 performance across different quality. So the results were very promising, like GMP, basic GMP, and REO grade all deliver consistent high gene editing efficiency in primary T cell. And this highlights that even our REO grade reagents are made with GMP-like process, but they are affordable for early research and easy to upgrade to clinical as your work move forward. And let's focus into advanced gene editing tool like base editor and the prime editor. Here's some data showcasing like base editor efficiency by using the adenosine base edit protein in the RNP formulation. And across the different dose, you can see a strong A to G conversion, and it's mean that it's capable to do precise and efficient editing, and and that protein can also able to do B2M protein knockout, and providing it's effective at the disrupting the gene function. And we have also like prime editing gene editing efficiency tested in HEK293 T cell, and we see that it makes the precise 5-base pair deletion exactly where we want into the HEK the cell sites. And importantly, this editing doesn't harm the cell viability, which means that the cell stay healthy during the whole process. And this demonstrates the prime editing can make accurate changes while keeping the cell functional. So at GenScript, we offer the high quality version of all the base editor protein and prime editor protein, and all of them is licensed from Dr. David Liu Lab, who is a leading researcher in this field. And these also available both into RUO and clinical grade format to fit your research and therapeutic need. So here is like shows the table, like what different kind of catalog protein we have available for carried out the CRISPR work. And we offer like range of validated catalog nucleus protein, and it's like Cas9, Cas12, even Cas13, and for prime editing on for base editing. All these are well validated and ready to use format. And we also providing like few free samples to test in your system. So because today we don't have enough time, I'm not going to able to show each and every detailed information and the validated data sets, everything. But if you need more information, definitely you can reach out to GenScript after this talk. Try to reach me. I will definitely help you to give more detail and even free sample if you are looking for. And so when you need like and not only catalog product of the Cas protein, but we are providing the custom nucleus protein service also. So when you're transitioning from discovery to preclinical or clinical stage, our RUO and GMP protein expression service deliver the high quality nucleus. All the reagents are manufactured under the strict GMP guideline. And in addition, GenScript provide the comprehensive support to meet the regulatory requirement. So far we have like more than 25 GMP batch of the Cas protein, and 5 of these have already received the IND approval. So 5 project was looking for IND application, and we support them and they get that approval. So now let's shift our focus. Another essential reagent in the gene editing is the guide RNA. This molecule is very important because it's directing the nucleus like Cas9 to the exact DNA sequence which need to be editing. And there are different type of gRNA use depend on their application or end use. So like it's maybe sgRNA, pegRNA, and so let's start with the synthetic sgRNA, so which are highly effective for precision gene editing, and whether it can be used for the knockout or knock-in by using HDR template. And to support this activity, we are offered 2 types of sgRNA. One is the easy edit sgRNA, another one is the safe edit sgRNA. Easy edit sgRNA is a cost-effective option that delivers the high editing efficiency and flexibility, while safe edit sgRNA is which is HPLC purified. It's giving a higher purity, reduce off-target effect, has minimum impact on cell viability. And all our sgRNA comes with the chemical modification to improve the stability and editing efficiency. So you can see, and it's also available from GMP grade to with the all QC requirement. So like we are providing extra QC also, like microplasma testing, endotoxin stability, even cytotoxicity testing in the customer's preferred cell line. So, and if you are working with the high throughput system, we can provide the sgRNA in 96-well plate format, so you can use them in the screening, and then further when you're going like preclinical or clinical, you can do custom service also. And to make the gene editing easier, GenScript offer even offline design tools. So what's exactly this helping to design the guide RNA as well as HDR template? And you just need to enter like target gene and the tool recommended for high editing efficiency and low off target effect. So it's automatically selecting the proper sequences, as well as one if we are looking for the tag protein for the deletion, detection, or purification, the option is there also, like one can select the different protein tag. And it's also can allow to pick the amount needed or purification specific method you want and how you want them in delivery format. So it fits to project perfectly to carry it out like sgRNA synthesis. So this is very unique, useful tools, and that's helped lots of customers to make their order through using this design tool. And next up is the pegRNA, which is play a very important role into prime editing. It's pegRNA not just telling where to edit, but it also carries the new DNA sequence to insert in that specific location. So it's because the pegRNA are very long and more complex structure with extra part, like here as shown, it has like PBS and RTT. So we launched like dedicated service for just pegRNA synthesis. And with over 20 years of our RNA expertise, GenScript can make custom PEG RNA up to 266 NT, means that's long big, with over 200 modification option. And here are the few examples which showcasing how editing efficiency of PEG RNA. So in this experiment, cells were transfected with varying dose of the 141 nucleotide PEG RNA, either into the desalted form or HPLC purified. And after 3 days, we analyzed the genomic DNA with Sanger sequencing, and we show that editing efficiency reached 52% with the desalted PEG RNA, and it's reached to 63 with the HPLC purified version. So importantly, the editing efficiency increase as the dose increase, and it shows like clear dose dependent improvement. And in the second experiments, we compared the GenScript PEG RNA with the competitor product using the PE2 and PE3 protein system. And both our desalted HPLC and purified PEG RNA match or outperform compared to other vendors in editing efficiency. So this confirmed that the PEG RNA deliver is reliable and competitive to the others. So it's like the good quality and can show use for the high editing efficiency, and it has the consistency. And for those running like pilot study or needing the quick validation, we also have like ready to use prime editing catalog product. So they are available in both desalted and HPLC purified format, and also offer like into the flexible quantity to match the different scale of experiments. In addition to our off-shelf product, we also offer like custom PEG RNA synthesis similar to like sgRNA synthesis service. And customer can select like whether they need the HPLC purified or extra QC option. All are available. So we have like built-in chemical modification to boost the performance along with the QC report. And in addition, we launch like new design tool just for the PEG RNA synthesis. So here like one can enter the gene symbol or sequence, and the tools walk you through the entire design and ordering process. So it supports the variety of edits like knock-in, knockout, sequence replacement, and it has also like interface, interactive interface, so it can easy to use and quite user friendly. So after like discussing about the Cas nucleus, which is one of the essential material, another one is the RNA. Now third one, we are looking towards the donor DNA templates. So this is also like very useful, and it's known as the HDR. So HDR template can introduce the specific DNA sequence at the targeted genomic location with high precision. And to maximizing editing efficiency, careful selection of HDR template is also essential. And GenScript offer like high quality HDR template in 3 different format. So one is the linear, another one are the circular. So let's start with the gene exact ssDNA, which is also known as a single standard DNA template. It has the ultra low in toxicity and ideal for sensitive application like T-cell therapy and generating the cell and animal model. And then we have the GenVen. It's a dsDNA. It's a close end, so it has like close end, and it's a linear format and has a higher stability and integration accuracy. So it's ideal for large scale knock-in, and it supports the insertion from 2 to 10 kb. And finally, we have like GenCircle dsDNA. It's a small circular DNA vector, which is free of antibiotic resistance and making it an excellent, so due to the absence of bacterial original sequence, it's an excellent non-viral alternative tools for knock-in or antigen expression. And this mini circular template comes only with 429 backbone, and which can help to boost knock-in efficiency up to by 30%. And because of that, it's also reducing the cytotoxicity and immunogenicity and antibiotic resistance risk. And it can support the insertion length below the 50 kb. And all these templates are delivered with high purity and with sequence verification and available from microgram to gram scale and from research grade to the GMP grade. So let's focus on the one experimental data carried out with the GenCircle dsDNA. So GenCircle dsDNA, it's like compact circle, which can also serve as a transpons vector for gene delivery. So in this example, like 3.5 kb CD19 CAR GFP knock-in construct was successfully introduced into T cell using the GenCircle dsDNA. And by using the MF transpons with the PGK promoter, high knock-in efficiency or approximately like 60% was achieved. And this demonstrates that here it shows like 60%. And this demonstrates like GenCircle system provide a highly efficient, stable, and scalable approach for transpons-based gene delivery, and seems it's valuable tool to advance cell and gene therapy application. So when we talk about the gene editing, it is not just the editing reagent that's matter. Its delivery format is also important. And the delivery format can really impact the editing efficiency, safety, even affect the regulatory process. So in recent year, like people moving towards the mRNA, because mRNA becoming more preferred as a non-viral delivery method, especially in the T cell based immunotherapy, or I will say immunotherapy, because it's allow efficient gene editing with minimum disruption of the gene. And to support all these new movement, GenScript offer the new range of off-shelf mRNA products. So these include like gene editing mRNA, so it's available into different format with different protein, including like Cas12, Cas9, even base editor, prime editor mRNA formulation. And this including the, it's also available like, along with that, we're also providing like reporter gene mRNA and reporter gene saRNA and reporter gene circular RNA. So it's available in all the three format of their RNA, like circular and linear. And we also have ladder to use them as a control while doing the experiments. So these products are reliable and research ready form and designed to help to maximize the success of the gene editing workflow, because they are off shelf, so you can get them quickly within 5 business days. And we also support large scale order, often used for bundling with other custom service. And that's way one can move their idea to success faster with confidence and with quality material and consistency. And as we know, like electroporation is a commonly used for mRNA delivery, but it's often cause significant cell death, particularly sensitive cell like T cell. So because of these, many researchers are switching to the lipid nanoparticle based delivery. and which are gentle, scalable, and increasingly preferred for clinical application. And even it can be used for the in vivo CAR T cell therapy. So to support all this shift, GenScript offer validated, ready-to-use, ready and edit LNP solution, which is designed for efficient and scalable delivery. And our off-shelf product cover the broad range of application, including Cas9 knock-in, knockout, as well as Cas12, and even like prime editor and base editing RNP formulation. LNP formulation. And since the LNP formulation greatly impact the editing performance, these catalog products are optimized for high efficiency and consistent results. We have like number of data sets available, but as I mentioned, I have very limited time to showcase each and every data sets. But definitely, if you are interested, we can share all that data with the paper and to make confirm that the quality and consistency is always there with each LNP formulation. And as the mRNA-based gene editing is advancing fast and researchers are looking for smooth end-to-end workflow, at the GenScript, we offer end-to-end solution from gene synthesis all the way to the LNP formulation. And this is like integrated approach. So that will give more control and high quality mRNA so that can bring high expression, and along with the each step you get the QC report. So that's like very good control process. And our offering including the wide range of mRNA. So that's including like IVT mRNA, circular mRNA, or self-amplifying RNA, and all are available for custom service also. So together, all these solutions or tools which we discussed, which support the entire gene editing workflow from early discovery through the therapeutic development, and helping anyone who is working in this area to advance their project faster, advance their idea into real world very quickly with confidence. So this is about the tools, but let's talk now how these tools make real world impact. So I will share only a few examples to show how these GenScript tools have supported gene and cell therapy program moving forward successfully. So let's take the example as a base therapeutic, and they have like NK510 program become the first FDA approval base edited NK cell therapy for solid tumor. Last December they get FDA approved. And GenScript supports this milestone not only by supporting the GMP grade sgRNA for their gene editing workflow, but also by assisting with their IND submission. And not only that, they also use our cytosine platform, which is a cell isolation technology, and including this nanobead and cytosine 1000 platform, which further supported their achievement. So similarly, another example is the UCLO. So UCLO Biotech, which recently received FDA approval for their core blood derived allogenic CD19 targeted CAR T cell therapy product. And this is also the first FDA IND approval for CAR T cell therapy from cord blood. And GenScript support this development by providing the key raw material, including like NSEED T cell activation reagent and cytosine cell isolation activation reagent and cytosine 1000 platform. So our nanoparticle-based isolation and activation technology, along with the regulatory support, help make this therapy more accessible to the many patients. So to further illustrate like our role in advancing the field, here are some examples of the partners using our high quality sgRNA and DNA templates. So one of them is like Biohang Biotech, which is using the GMP sgRNA to support their UCAR-T IND approval. And another one is the BRL medicine also use our sgRNA, and they also get like CAR-T IND approval globally. And then the Maximum Biotech, which is also used sgRNA services for CAR-T cell therapy. Similarly, for the Neogene therapeutic, they developing TCR edited T cell, and they use like our DNA template, which is for carried out the T cell therapy. And the last but not least, like which is editor's medicine, which they also achieve over 90% knock-in efficiency using our non-viral DNA template in their Slick platform. So all this reflect like GenScript commitment to the quality, consistency, and on-time delivery made us as a trusted partner among many in gene and cell therapy development. And so far, we deliver like more than 130 clinical batches, which is supporting more than 50 IND submission, and 13 program achieve global IND approval. So, and there is many more it's happening in the pipeline. So this shows how GenScript is supporting the people's idea to bring into this real world. And to at the end, I like to say like cell and gene therapy is advancing rapidly, and success is highly depend on high quality reagent, effective tools, and partner who truly understand both the science and regulatory landscape. And in this area, GenScript provide like comprehensive integrated solution to support from discovery to all the way through development. And if you like to learn more about the automation platform or other tools which we have, so please don't hesitate, reach out to us via email. And this brings me to end my presentation. And thank you for joining us today. And if you have any question or would you like to discuss for your specific project or need, please reach out to us. Thank you everyone again.
Thank you very much, Dr. Patel, for that presentation. We are running a little bit short on time, so in the interest of moving us back on schedule, I was just wondering, what is the best method for anyone interested in what GenScript has to offer to reach out and gain additional information?
Yeah, they can send us me email, and then we can start communication from there. So that's where our sales people and then field application scientists also can understood the need, and we can provide immediate support.
All right. Thank you very much for being with us today, and thank you very much again for that presentation.
Thank you. Thank you, everyone.
Our next speaker is Carrie Eckert, who is the synthetic biology group leader at Oak Ridge National Laboratory. She previously held positions at the National Renewable Energy Laboratory and the University of Colorado Boulder. Eckert's work focuses on developing CRISPR-based methods for genotype-phenotype discovery and metabolic engineering in microbes. Her research aims to accelerate the genetic manipulation of eukaryotic and prokaryotic systems for biotechnological applications. All right, Dr. Eckert, let's see if we can get your presentation up. Wonderful.
There we go. Oh, let me do both. So can everybody hear me? Yeah.
I can hear you and I can see your screen. Perfect.
Right.
Please, the floor is yours.
Thank you very much for the invitation. So really what we're focused on, and we're all funded at the national labs under the Department of Energy in some ways, and we're really kind of pushing towards the circular bioeconomy. What does this mean? It means utilizing biology to make products from waste and sustainable feedstocks and be able to make that very circular. So be able to use all the things and make all the things more sustainable and more usable, worthwhile. And we really think synthetic biology is critical for this. So the ability to actually engineer performance advantaged plants and performance advantaged microbes, and as well as other technologies involved in that. But synthetic biology is really at the center of this. So synthetic biology is really pushing the limits of organisms. So we're really fighting against natural evolution and the organism's ability to survive versus what we want to engineer, like improved traits, like yield and desired traits, like product production of a particular product. We're really limited by our gaps in knowledge. So genotype to phenotype relationships and complex network interactions. So even a very well-studied organism like E. coli still has about a third of its genome that we don't know the function of. So we really are trying to get at this. And we're also going to be affected by our ability to effectively transfer complex phenotypes. So things like feedstock utilization, so the ability to fix CO2 as a feedstock, that's not easily transferable, at least efficiently. And we're going to also be affected by the available engineering tools. So this really goes down to engineering and modeled organisms. So everybody knows about E. coli and Saccharomyces cerevisiae. They're very commonly used in industry as well as a lot of research. There's great genetic tool sets and foundations of physiological knowledge, but they lack a lot of the traits that we're going to need for a broader range of industrial applications. What we really like to do is start with nature's best for these processes. So, things like tolerance to different compounds, their ability to grow on different feedstocks. And so we really our focus is on development of these non-model organisms and developing of genetic tools for them. And so I have a team of researchers that I oversee at Oak Ridge National Lab. They're all fantastic, but we're really all involved in developing these tools for non-model organisms to really advance the bioeconomy. So everything from bacteria to fungi to plants, we want to be able to engineer them in many different ways to actually advance our bioeconomy. So there's really three main barriers to genetic engineering of novel organisms. You have to be able to introduce the DNA into the cells. You need to have that maintained, and you also need to have that functional. So we're really developing workflows across these. We have to first get that DNA into the cells, and so a lot of this is a lot of trial and error and trying different things. But once you kind of have that in place, one of the things that's really a problem in bacterial systems, so not just CRISPR as an immune system, but there's also immune systems that involve restriction modification systems. And so this is where you have unique sequences of methylation where these bacteria can actually tell self from invader. And so we've been developing systems at Oak Ridge, and this is with Adam Gus and Bill Alexander, where we can actually use Oxford nanopore sequencing to look at the methylation profiles of the DNA. We can actually mimic that in our cloning strains of E. coli, and this really allows us to get that DNA into the cells. So just kind of a quick overview of something where we've actually implemented this is looking at this Bacillus velezensis. There's a couple of different strains of this. This is actually a commercial biostimulant called Companion, and it's used to reduce fungal pathogens. But one of the main problems is we can't get it to establish in soils and stay there to really protect the plants. And so what we did is to develop these genetic tools. We put it through this pipeline. We did the Oxford nanopore sequencing. We were able to look at the methylation profiles. And if you look at the graphs here, so this is just plus arabinose means when we're inducing those methylases, you're able to greatly increase transformation efficiency. So this allows us to develop other tools like our ability to integrate genes into the genome, looking at CRISPR-based tools, as well as a serine integrase system where we're able to introduce heterologous genes very quickly and efficiently. So really in this design, build, test, learn, we need to be able to engineer optimized organisms. And what we need to do that is all the parts and pieces in there. So you need all the promoters, ribosomal binding sites, other elements, but we're really here, I'm going to focus on what we're trying to do in terms of introducing CRISPR into these systems, both for engineering as well as functional genomics. And so what is functional genomics? This is our ability to link genotype to phenotype. So most commonly people are doing adaptive laboratory evolution. This is where you're just doing serial dilutions of these microbes over time, and you're growing them under stress conditions, so they're acquiring mutations, and eventually you'll have mutations that are acquired that actually improve their ability to grow. This is actually really easy because it's simple. It covers diverse fitness paths, and it's not limited to genetic tractability, but it's very slow. You know, imagine with these serial dilutions, this can take months to actually be able to get at the actual genetic change. And it also can be really hard to determine the gene to trait relationship. As the mutations are accruing, they're eventually going to level off. So you're going to have more mutations than mutations that are actually improving growth under those conditions. There's also the use of transposons with sequencing, where you're able to introduce transposons into cells, and they'll randomly insert into genes. And you can apply a barcode to those, so you can identify the causative genes following the selective conditions. This is limited to non-essential genes. So you imagine you have an essential gene in the genome. You can't use this method because it's not going to be able to survive. And it can also be very difficult to generate these libraries where it's covering all the genes. But it can be very useful to identify gene targets. We're really moving to using CRISPR-Cas-based methods here. So here, if depending on your system, you can utilize targeted editing, the CRISPR interference for knockdown or activation in a genome-wide, easily tractable system where we're actually able to track each mutation by doing next generation sequencing of those plasmids that we're introducing that have unique guide RNAs or editing platforms. But this is where we're really limited by available genetic tools. So we have to have different things like inducible promoters, and we have to have decent editing efficiencies. So this is really where we're coming at using the CRISPR systems. And so in bacteria, they don't have non-homologous end joining that people have been talking about, where we can't just disrupt gene function. So we're really focused on homologous recombination if we're doing editing, but we can also do CRISPR interference or activation. And in our systems, we focused on CRISPR interference. This is the simplest. Activation tends to be a little difficult in bacteria, but we've got interference working wonderfully. So we are basically generating CRISPR libraries that are affecting all the genes in the genome. So we design CRISPR guide RNA designs for 10 to 15 guides per gene, and this allows us to have a variability of knockdown. And we're also doing this with inducible promoters, so we can actually induce that knockdown, so we can see both essential genes and non-essential genes. We order these as oligo libraries. We assemble them using Gibson assembly into plasmids, and then we transform them into cells that have an inducible dead Cas9. And then we're able to, after the selective pressure, we can look at depletion and enrichment of the population before and after the selective condition. So here I'm going to talk a little vignette about what this was work that was started when I was still at the University of Colorado Boulder, but we were focused on a cyanobacteria called Synechococcus 7002. So we really wanted to look at the effects of this guide RNA library under different light and temperature regimes, excuse me, because light and temperature can induce stress. And these organisms are very important to this planet because they're the biggest photosynthetic organisms, more than plants, because they're in the oceans. And they're really affecting by daily fluctuations in light, by differential temperatures, and also the spectral quality. So the light as you're going deeper into the water. And light and temperature really, they reduce growth rate. So you see in here is on the top are just normal, more permissive conditions. Even with red and blue light, you still see growing very similar to a wild type. But when you're putting these under really stressful conditions, so cold temperatures with red and blue light, it greatly decreases the ability for these cells to grow and divide. And so here, what I'm going to show, and this is going to be indicative of the graphs that I'm showing here, is so each guide RNA is representative around this circular chromosome structure. So this is a Circos plot. So for every gene, you're able to see whether that particular guide is depleted or enriched under a particular condition. And so in this case, we have about 33,000 guides going around the genome. So here is just under normal permissive conditions. So white light under 37 degrees Celsius, and you'll see most of the genes are going to have a reduced fitness. You're really the only things you're seeing that are improving fitness are the circadian clock proteins, because here you're growing under 24-hour white light, so they're not needed under these conditions. But we have gut checks of like ribosomal proteins. They're highly decreased in enrichment. So we really know that this method should be working, and it aligns really well with T-and-seq studies that we see in different cyanobacteria. And just in general, in white light standard temperature conditions, there's very minimal differences. Most things are depleted. There's a few things that will stick out that are enriched. But what we did was we combined the cold and the red light, blue light, and what you see is this huge increase in the number of those actual guide RNA repressions where you're able to see a large enrichments under those conditions. And really, are these fitness mechanisms similar between the cold, red, and blue? So they're the same guides, but we see very diverse responses, and they're very different from what you're seeing in the more normal conditions under 37 degrees with normal light. We were able to group these into different clusters. So looking at things that are major depletion, minor depletion, red or blue enrichment, or just generally enriched, and examine the top 3 gene ontology categories within each cluster. And really, there's a real lack of major overlap. It's suggesting that there's unique stress responses and alleviation mechanisms in both the red cold and blue cold conditions. And really, a lot of these genes that we're seeing that are actually enriched in growth are actually essential genes. So these are things that you would never see in an RBT-and-seq study because they would never show up because they're essentials. One of these are a lot of the different genes involved in NDH1, which is an important conserved complex that's involved in cyclic electron flow and respiration and photosynthesis. There's also particular subunits that are associated with inducible carbon dioxide uptake or constitutive carbon dioxide uptake. And what we see is the degree of enrichment and the presence of enrichment. There are distinct alleviation mechanisms. So under 22 blue light, there's actually reduced constitutive CO2 uptake that's advantageous. We're also able to show that the fact that we're using 10 to 15 guides per gene, the optimal level of repression is condition specific and variable. And so what you'll see here, this is for NDHC on the left, is these are each representing a guide. And so you go from things that are their decreased growth as well as improving growth. And those are different across the 22 red and the 22 blue conditions. And we actually were able to pick things, these guides that are low, medium, and high knockdown that associate with low, medium, and high growth. And we were able to show that we had transcription rates that were mirroring that, as well as we were able to induce the expression using IPTG at different levels to show that this is the case. We're also able to show that mismatch CRISPR interference can increase density and map gradients. So we're actually able to make some mismatches in that guide RNA to the target sites. And you can actually look at that very mismatch sensitivity and differences in log fold change of growth. So really, a lot of these stress mechanisms of cold, red, and blue have to do with the photosystems. So you're looking at the photosystem 1 and 2. They're affected differentially by the red and the blue light. And what we're actually able to see that in the photosystem ratios. So the ratio of photosystem 1 to photosystem 2 is greatly different in the 22 blue conditions. And we also see a difference in the phycobilisomes or the light harvesting structures between the blue and the red, as well as white lights. So these native acclimation routes are actually inadequate. They're maybe even overcompensating with the overexpression of these things. So knockdown is actually alleviating some of that stress. And so the cold temperatures are exaggerating the stress from energy imbalance. So the red and the blue light alone, you don't really see these, but when you're adding the cold, you're really bottlenecking things through that plastoquinone pool. You're reducing the ability of the cells to actually control this electron transfer. So with those knockdowns, we're actually able to improve those growth. We also came up with a mechanism to try to look at 2 gene knockdowns with the dual guide system. And we actually utilized a native RNA hairpin that is cleaved by RNase 3 to be able to build these. And this is on the right is just showing phenotypically, we're able to take, this is part of the phycobilisome, and it's a colorimetric thing, so it's affecting its ability to fluoresce under different light conditions. And you're also looking at FTSZ that affects its ability to divide. And so we're actually able to show phenotypically when you add these two together, you can see they're both knocked down together. But when we do this with some of the library components, we're actually showing that these fitness effects are nonlinear. So just because one had a positive effect and the other had a positive effect, together they generally don't. So what we're showing is these fitness effects are really non-additive. So it's like there's going to be some counterintuitiveness in actually combining guides in this ways. So really here is a take home that many essential genes can be partially repressed to improve acclimation to stress conditions. The amount of repression is going to be variable under these, and it's really dependent on your gene target and the growth condition. And combining stresses can actually cause imbalance in the photosystem excitation, and the double gene knockdowns have really unexpected behavior. So shifting gears a little bit, we're applying these methods to other systems. So this is a project, the Center for Bioenergy Innovation. So this is a bioenergy research center that's funded by the Office of Science, BER. So the Biological and Environmental Research Agency. And so these are very large projects. There's 4 of them across the country, that CBI being one of them, led out of Oak Ridge, where we're looking at the whole system of being able to make different biofuels. So in this case, looking at process advantage feedstocks, so focused on native perennial plants like poplar and switchgrass, trying to improve their yield and resilience under different conditions. And then also going towards the microbial side. So on the left-hand side, we are utilizing an organism called Clostridium thermocellum. Why this organism? Because it can actually bind to ground up biomass. So poplar, switchgrass, you grind it up, it binds to it, it's able to break down the cellulose and hemicellulose components of that biomass and convert them directly to sugars that it ferments. So from this, we're making ethanol in this process. But on the other side, we're working with engineers, and they're working on using reductive catalytic fractionation to basically extract the lignin oil, and it leaves those solid carbohydrates that you can then feed to Clostridium thermocellum to convert into ethanol. But you can also do direct hydrodeoxygenation of that lignin oil to go towards cycloalkanes and other aromatics that can be used, as well as using a biological process using an organism called Pseudomonas putida that's able to directly convert those lignin aromatics into products of interest. And our goal here is really to make a fully sustainable aviation fuel. So all parts of this are coming from sustainable sources. We've actually deployed the CRISPR interference enrichment and depletion libraries in Pseudomonas putida. So this is a bit larger than the cyanobacteria library at about 33 to 34,000 members. This is almost 79,000 members with 15 guide RNAs per gene. And we're growing these under conditions that can be really toxic. So coumarate and ferulate are components of the lignin aromatics. They're common ones when you're doing breakdown, but at high concentrations, they can still be toxic, even though the organism can utilize them. So we are now in the process of evaluating these libraries for Pseudomonas. We're also working to develop CRISPR interference systems in Clostridium thermocellum. So the big issue here is this organism is a thermophile. So it's a strict anaerobe as well as a thermophile. It is not easy to work with. And what's important about it being thermophile, none of the tools that have been developed that have even been talked about today actually function in it, because they only function at mesophilic temperatures. They won't function at temperatures above 50 that this organism grows in. So we really needed to, number one, identify some inducible promoters so we can control that CRISPR interference systems. And we had previously found CRISPR systems that function in Clostridium thermocellum. One of them is a Geobacillus Cas9 that we use for editing. that was previously found in Jennifer Doudna's lab. We were able to show that it functions in Clostridium thermocelum. But we also characterized its native cascade system. So this is a type 1b system where we wanted to use this one for CRISPR interference. So we can delete the nuclease component Cas3 and put that under an inducible promoter, and to be able to develop these libraries. So we're actually in the process of developing these libraries for Clostridium thermocelum. What we want to move towards in these non-model systems is going towards editing. So being able to make direct edits in a library format to really dig down to amino acid level of what the function of some of these proteins are under different stress conditions. So this is a platform technology that we developed at the University of Colorado Boulder. It's called CRISPR-enabled trackable genome engineering, or CREATE. And so this is where you take a sequence. So you identify a PAM mutation. So this is the SPCas9, where we're making a synonymous PAM mutation. So just making a mutation so that the CRISPR-Cas machinery can no longer recognize that region of the genome once an edit is incorporated. And we're linking that to another edit of interest. So in this case, just making a stop codon. But we found that if you make the PAM site too far away from that integration site, the sweet spot is about 17 base pairs, you start to incorporate the PAM mutation, but not your mutation of interest. So using those design rules, we're basically building oligos of in this case, 230 base pairs, where we have the synonymous PAM mutation, your homology arms, as well as that your mutation of interest, and then a guide RNA expression for that particular guide that's going to recognize the region in the genome. And these can be used to build very large libraries. So we're using basically oligo printing that you can do at Agilent or GenScript or other places. And we're able to just build these into the plasmids, similar to what we were doing with CRISPR-I. In the case of E. coli, we were able to build up to a little over 110,000 member library, and Saccharomyces, we did about an 83,000 member library. And we're able to interrogate these in a pooled fashion, so growing them under selective conditions, similarly to what we have done in CRISPR interference, to identify unique mutations that lead to that, some of which are known in the literature, some of which are not, because we're able to make mutations that don't really happen naturally. And so this is really going towards being able to validate some of the mutations that can be used to engineer more robust organisms for production and tolerance. At Oak Ridge, we've also been using this technology to start building essentiality libraries in E. coli. So what this means is we've targeted all the essential genes in E. coli, and we've done alanine scans using the CREATE technology. And we're able to find individual amino acids in each of these genes that are either depleting or enriching growth. And in some cases, they do enrich growth. And so we're now working with a researcher at Oak Ridge, Erica Prates, who's helping us to pair with predictive models utilizing AlphaFold to start to really get to the amino acid essentiality, I guess, essential of E. coli that we can now start to utilize to help improve predictive models in general. So we've been working on improving CRISPR-Cas9 editing in Pseudomonas putida as part of CBI. So we've got CRISPR-Cas editing working well, but the major limitation is the homology arm length. So once we drop down from about 250 base pair homology arms, the editing efficiency drops quite precipitously. We need these to be shorter to be able to use the CREATE type methods. So we're right now trying to do some active projects where we're trying to reduce that homology arm by using different recombinases and really moving towards being able to do CREATE style studies. But we're in the meantime kind of developing some intermediate methods to be able to do some of these mutagenesis assays. So another project we're actually utilizing this for, utilizing Pseudomonas putida as well, is under the Bottle Consortia. So this is a joint project with the National Renewable Energy Laboratory led by Greg Beckham. And this is work with Adam Gus, that's also at Oak Ridge. Really looking at utilizing Pseudomonas putida for plastic upcycling. So plastic parts, they look very similar to the lignin. You can actually engineer different enzymes that are able to break down the plastics and be able to engineer the organisms to make products of interest. And so working with chemists, they found that replacing the alpha hydrogens and PHAs with methyl groups actually improves polymer properties, but this is really hard and expensive to do chemically. So our goal here is, can we biologically produce these precursors as a precursor towards better PHAs that can be synthesized, that have better properties? There are no known biological routes, but there are enzymes that look very similar to the different steps of this. And so what we're doing is basically a de novo pathway building using retrobiosynthesis. And what we want to do is a combination of ALE, adaptive laboratory evolution, along with a create style system to be able to evaluate better enzymes to do this process. And then lastly, I'm going to kind of move into what we're looking at next. So we're developing these CRISPR tools. We now have pipelines to get things to work in a lot of different organisms, but our automation tasks are really a bottleneck. So once we get to that point of we now have all this amazing data from these CRISPR interference libraries or editing libraries, going through to validate what's actually happening in those individual organisms with those individual mutations is hard. So we're wanting to use automation to really amplify and speed up our build and test parts of the design, build, test, learn cycle. And so at Oak Ridge, we've actually just onboarded 2 TCAN Fluent systems to do this. So one is on the left is aerobic, and we have actually one that's fully anaerobic. So this is enclosed in a massive glove bag that is under anaerobic conditions. So this is allowing us to do high throughput electroporation and conjugation, liquid handling, dilutions and plating, as well as incubation and shaking, colony picking, and it also has incorporated microplate readers to be able to track growth. And really, moving forward, we're hoping these can really help advance some of the research that we're doing. And so this has been spearheaded by a great postdoc, Andrea, and she has lovingly called these, on the left-hand side we have Kirk, and on the right-hand side we have Spock. So we're really excited to get these going. And lastly, we'll just have a couple of neat videos because she's nerded out over this, but how we're actually able to we use a Lonza 96 well electroporation system to do the electroporations, and then we're doing dilution platings of those so we can utilize fewer plates to get to individual mutants. So we're really excited moving forward. And then also lastly, this is really a cross-disciplinary effort. We need to be working with a lot of people. So I really want to start to take some of these data sets we're generating and work with computational biologists to feed better predictive tools. We're also working with engineers in this space, and we're also working with TA, LCA analysis, so techno-economic standpoints. If we want a particular organism to have a certain process, is it even worth starting on? So really trying to narrow what we focus on to get to being able to industrialize things a lot faster. So with that, I will thank the funding agencies and all my collaborators, and I will stop for questions.
Thank you very much, Dr. Eckert, for that presentation. As we begin our Q&A session, I wanted to ask, you mentioned early on that one of the obstacles to moving into CRISPR-based gene editing or CRISPR-based applications is a lack of tools. And I'm curious, how do your tool needs and priorities, how are they similar to and how do they differ from say for the priorities for basic health research or healthcare or diagnostics or those sorts of things?
Yes, some of them are going to be similar. Obviously, that's one of the great things about CRISPR is its applications across a variety of systems. And I think, suffering from some of the same problems is getting things to edit efficiently. So some organisms are really good at homologous recombination and others are not. So it's really, the key points are all going to be similar across things. I mean, obviously they're going to be specific to the organism, but being able to express genes, being able to insert genes, being able to delete genes, all of those things matter. So I think there's a lot of parallels, but bacteria grow faster and are a little bit easier to manipulate from the start. So I think that helps a little bit, but some of these organisms are definitely a lot more difficult than others.
Thank you very much. You also talked about challenges of developing editing programs, or sorry, editing platforms for some of these models. And I'm curious as to how much is potentially applicable across different model species, and how much has to be tailored towards an individual species.
Yeah, so there are things that do have to be tailored. So you're going across different bacterial genuses. So you have things that are gram-positive, gram-negative. The promoters are different. What we're actually working with is, so Adam Gus had developed this serine assisted recombinase system to be able to drop things easily into the genome. So you can put a landing pad in there, and once that's there, you can start to evaluate the different parts and pieces. And so we're basically curating libraries of promoters and other genetic elements we want to study in those organisms and utilize, so we can quickly evaluate which ones work and which ones don't. So to get to those answers faster.
Thank you very much. And I think one final question to wrap up the Q&A. The gene repression experiments, and I was curious, is there a way to gain some sort of predictive insights on what could potentially happen there, or is it mostly repeated experimental trials?
Some of it can be kind of inferred and predicted from what's known in the literature, but some of it is absolutely novel. And I didn't talk about this, but a large majority of those genes, especially in Pseudomonas putida, a large majority don't have an annotation. So we don't know what they do. So this is a point where we're now starting to work with some colleagues doing large language models using machine learning to start to see if we can't interrogate the data sets, especially when we're able to grow them under different conditions. Can we start to align in things and group things in areas where we have a better understanding of what they might be doing, and start to help use this to build better predictive models, just to amplify our ability to decide which ones we want to focus on and validate.
And as a kind of a follow-up to that, what is our familiarity with the genomes of these organisms in terms of data available, the depth of data relative to what we understand about the human genome?
That really varies on how much they're studied. So Pseudomonas putida has kind of arose. It's now, I would say, technically a model organism, and the same with some of the cyanobacteria used just to study photosynthesis in general. A little bit more is known, but you go into things like Clostridium thermocelum. It's a very unique beast. We've been working with it for a number of years, but there's still a lot we don't know about it. So it really just depends on how much research has been done on some of these before, but some of them we can just pull out and they're completely novel. So we kind of have to start from scratch. That's really why we want to develop some of these methods, so we can get to some of the answers quicker.
All right. Thank you very much again for answering those questions and for your wonderful presentation. With that, let me introduce our final speaker for today. Eric Sontheimer is the Pollard Chair in Biomedical Research and a professor at the University of Massachusetts Chan Medical School, where he also serves as Vice Chair of the RNA Therapeutics Institute. He has made foundational contributions to our understanding of RNA molecules and gene regulation, and in 2008, his lab reported that CRISPR systems functioned via DNA destruction. He co-founded Intellia Therapeutics in 2014, and continues to drive research on CRISPR-based therapeutics. His many leadership roles include co-chairing the Board of Scientific Counselors at the NCI and serving on advisory boards for biotech companies.
Thank you very much for that introduction. Is my screen visible?
I can see your screen, but the presentation is not full screened. There it is.
Is that good?
Yes.
Okay, great. Thank you for the kind introduction and the invitation to give this talk today. The title of this talk is Precision Insertion of Multi-Kilobase DNA Fragments via Prime Assembly. And a few disclosures are shown on the introduction slide. So we've already had a bit of introduction from the previous speakers on this, but CRISPR, of course, has been repurposed as a revolutionary genome editing platform. And there are three primary modes of CRISPR-based applications for this. The first is that of nucleases, where it can introduce either double-strand breaks or single-stranded nicks into defined sites of the genome in an RNA-guided fashion to induce repair events that can edit the genome. In 2016, David Liu's lab reported on base editors in which a nickase Cas9 is fused to a deaminase that can drive single base changes in targeted sequences. And then again, David Liu's lab in 2019 reported on prime editors, which use a fusion of nickase Cas9 and a reverse transcriptase to introduce templated changes, user-defined, into the genome. And these last two forms of editing are examples of precision editing, where you are not relying on difficult to control repair outcomes to the same extent, but rather you are specifying in advance exactly which sequence change that you wish to generate in the genome. And of course, there is great utility from just introducing indels by double-strand breaks, but there's even more utility from doing precision editing to make specific changes in the genome. And much of the effort of the field in recent years has been involved in trying to develop new precision editing platforms, increase the efficiency with which the existing ones function, and increase the sizes of the edits that can be introduced. So prime editing will be the topic of my presentation today. And so just by way of a bit of background about the pathway, because this will be important, you have a nickase Cas9 and a reverse transcriptase. And then in green here, we have the PEG RNA, which has a 3' terminal extension with a primer binding site and a template region. And so you have the RNA-guided nicking in the usual Cas9 fashion, but this is where the 3' extension comes in, where now you can anneal to the primer binding site of the newly generated nicked site on the DNA, and the reverse transcription template is immediately adjacent, and the new nick can be a priming site that can lead to primer extension that now specifies the sequence that you have pre-specified in the template region. And so now if we strip away the editing machinery, we are now relying on endogenous repair machineries within the cell. You have generated a 3' flap that carries the new sequence. That can spontaneously equilibrate into a 5' flap that generates a heteroduplex involving one strand which is edited and the other strand which is not. The repair pathway can then fix that in a way that incorporates the precise DNA edit, although it can also get rid of the newly edited strand and simply regenerate the original strand. To try to bias that outcome, a separate approach called PE3 involves a second single guide RNA that introduces a nick on the non-edited strand and biases the repair pathway towards the incorporation of the new sequence that you specified within your PEG RNA template region. So when this came out, it generated tremendous excitement in 2019 and since. It does not require double-strand breaks, which is an advantage in terms of potential genotoxicity. It does not require a homologous directed repaired DNA donor. It can edit non-dividing cells. It can introduce any substitution, not only those that are amenable to base editing, and it can also introduce indels, so it's significantly more versatile than other previous platforms. But there are a number of challenges associated with implementing prime editing as well, including but not limited to the following. One, the effector protein is very large, which makes production and delivery very challenging. The 3' extensions to the PEG RNAs are susceptible to degradation. There are some unpublished approaches, including ePEG RNAs and law protein fusions that help with this, but it remains an issue. The primer binding site of the PEG RNA and the guide sequence at opposite ends of the molecule are inevitably complementary to each other, which can cause misfolding and compromise loading into the effector. in practice, editing efficiencies are highly variable and often low, and that gets worse as you try to go to longer insertions and overwrites. The reverse transcriptase can sometimes read through into the tracer RNA region of the pegRNA, leading to some frequency of editing errors. And then in addition, it's very common to observe unwanted products from insertion or deletion mutations from repairing the flap structure during the editing process. So we and others in the field have been trying to take some of these challenges on. We first got started with a couple of them, but including the potential issues with the pegRNA. So just to reiterate, 3' truncation is a well-documented problem with pegRNAs because the single guide molecule is protected by the Cas9 protein, but the free PBS and RTT is generally not, leaving it susceptible to exonucleases. The self-complementarity of the spacer and the primer binding site causes misfolding. Tracer readover by reverse transcriptase can impinge on the scaffold sequence. And the self-complementarity portion of this was analyzed in considerable detail by Scott Wolf's lab and shown clearly to be a liability for the function of pegRNAs, leading to a search for potential solutions or approaches to mitigate this and other types of inhibitory difficulties. And I should also note, we just heard some very impressive work from our colleagues at GenScript on pegRNA synthesis. It is nonetheless true that these are very long from the standpoint of inline RNA synthesis by chemical means. The shortest pegRNAs are 125 to 130 nucleotides long. And at that length, even then, pegRNA synthesis is very challenging, especially at scale. And of course, the field wants to get to longer and longer edits. And the longer you go, the greater the challenge is. We commonly do bioanalyzer runs of pegRNAs that we obtain, and they are definitely not fully pure. And this remains a challenge, and that will become especially true as we try to go to clinical applications where the demands are especially stringent. Okay, so published work from 2022. This is a lengthy story, and I won't go through exactly how we arrived at this system, but we and then independently Keith Jung's lab showed that prime editing does not require necessarily fusion of many of the components. And in particular, the pegRNA can be split such that it once again relies on a canonical single guide RNA and a separate molecule we refer to as a prime editing template RNA or a PET RNA that can function separately. And we can tether it to the reverse transcriptase using an MS2 coat protein fusion. And furthermore, we can have the cell produce this PET RNA for us in circular form, greatly limiting the degree of exonuclease susceptibility that these things can suffer from. And so this was collaborative work with the lab of my colleague, Wen Xue, at UMass Chan Medical School and the RNA Therapeutics Institute. And it was spearheaded by two postdocs, Bin Liu and Xiaolong Dong. And a couple of things that we liked about this system: the smaller components can potentially ease production and delivery. It can simplify the development of new nickase RT combinations. The PET RNAs can circumvent pegRNA misfolding or instability or both. The nicking and writing modules can be independently varied and controlled, and perhaps additional advantages as well. So we then went on, having developed, gone from canonical PEs with pegRNAs to the split PE system with the circular PET RNAs. And of course, it was not difficult for us to recognize that circularization is not the only way to stabilize an RNA molecule for its function as a primer binding site and a template. There are many years of development of chemical tools for synthetic nucleic acids made by solid phase synthesis that can also be used to greatly stabilize and increase the efficacy and the in vivo compatibility of small nucleic acids. This, of course, comes predominantly from the antisense oligonucleotide and siRNA fields, and many of those lessons learned from those fields can be applied to CRISPR genome editing as well. And so we showed that we can make linear prime editing templates, or LPETs, again with an MS2 fusion to support prime editing using synthetic templates that are made in vitro and are more compatible and readily applicable to some delivery formats such as LNPs. Okay, so whenever we developed this, now the fact that we can readily make these as separate template molecules by chemical synthesis, it also occurred to us that we could replace the ribonucleotides that typically support reverse transcriptase function in the template region by 2' deoxy residues, or DNA residues, in the template region. And the reason that we were interested in doing that is because that would open up the door to applying a new writing module to the prime editing related pathways, in the sense that we can then replace the reverse transcriptase with a DNA-dependent DNA polymerase. And we took this system and made that adjustment, incorporated DNA residues into the template region, and substituted the reverse transcriptase with a well-studied DNA-dependent DNA polymerase called Phi29, which is the product of a phage. And the reason we did this is because its biochemistry is very well understood, and it is known to have proofreading activity that contributes to an orders of magnitude greater replication fidelity than what is observed with non-proofreading reverse transcriptases. It also has a DNTP affinity that is orders of magnitude higher than that of MMLV reverse transcriptase. And it has been shown in published work from both Scott Wolf's lab and Dan Bauer's lab that DNTP affinity can in fact be a limiting bottleneck in prime editing efficiency in cells and likely in vivo. Phi29 is also far more processive in its synthesis than reverse transcriptase. Phi29 can go tens of kilobases from a single priming event before stochastically falling off. And so all of these, we think, will contribute to, through further development, to a system that might have considerable utility and advantages over RT-based systems that function in canonical prime editing. And this was published as an EPUB in 2023, and then formally in 2024. And I should also just note that similar systems involving DNA-dependent DNA polymerases were developed independently and reported by the Kleinsteiver lab, also in Nature Biotech in 2024, and most recently by Piyush Jain and Cameron Mervold in their labs as a preprint last year as well. Okay, so I also just want to note one unanticipated issue with reverse transcriptases in addition to those that I just recounted in the previous slide. Wen Shui's lab has spearheaded an effort that showed, perhaps somewhat unexpectedly, that reverse transcriptases, including those that are associated with prime editing and including in the form where they're fused to nickase Cas9 as a prime editor, can actually change the outcome of double-strand break repair when present in the same cell. And so in this case, the double-strand break is introduced by Cas9, but of course, spontaneous double-strand breaks are known to occur as well. And as you can see, Cas9 alone gives rise to a certain indel spectrum at this particular target site, as shown by deep sequencing. But then when RT is co-expressed either alone or as a prime editor, specific indel products, in this case a CT insertion, increase dramatically. And then if you do the same, but with a negative control reverse transcriptase that is catalytically inactivated, it goes back to the canonical repair spectrum, and this was shown to be true at multiple sites. Similarly, but orthogonally, the manuscript reported that you can use laser microirradiation to introduce DNA damage. This is work that had been reported by others, and it was known that DNA repair factors, most notably PARP1, but certainly others as well, are rapidly recruited to sites of double-strand breaks to help initiate and orchestrate the repair process. And interestingly enough, when you take a reverse transcriptase, in this case MMLV, but also TF1 from some of the PE6 systems, when you fuse it to mCherry and follow that by time-resolved microscopy, you also see the rapid recruitment of reverse transcriptase to the same sites of double-strand break in those cells. And so this is potentially a source of genome alteration that is unintended during prime editing outcomes at sites of spontaneous DNA, double-stranded DNA breaks that we need to be watchful of and potentially account for. And we are actively pursuing the question of whether DNA-dependent DNA polymerase systems have the same effect, or is this another advantage that they might have over reverse transcriptase systems as well? Okay, so with that, I'm going to change gears and talk a little bit more about systems that the field is eagerly pursuing to try to generate large insertions. This is something that was touched on by Ben Kleinsteiber in his talk earlier today, because if we can overwrite entire exons or even entire genes, or in some cases for recessive loss of function alleles, if we can insert a single copy in an advantageous genomic locus of a gene, then we could have individual editing configurations that could be applied to many different pathogenic mutations. And there are many inherited diseases where there are not specific alleles that predominate among the patient population, but rather a large spectrum of mutations that collectively cause disease, but individually are quite rare. And so to be able to address an entire population of pathogenic mutations is a major goal in the field. And so a couple of platforms. Now, of course, we can do this by homology-directed repair in cultured cells, and in some instances in primary cells as well. But it is difficult to implement, it does not scale, and it's largely incompatible with most in vivo applications. And so that is not likely to be a long-term solution. So multiple labs are developing CRISPR-associated transposons, or casts, and trying to engineer or evolve them to have greater efficacy and accuracy in mammalian cells. Prime editing systems have been combined with integrases and recombinases to use prime editing to introduce specific recognition sites for integrases or recombinases that can then be BXB1 being a classic example, that can then be used with a donor template to drive the insertion of a gene-sized cassette at single copy into a defined site in the genome. And these are also being actively pursued by many groups. And then, I could keep going, but suffice it to say that there are people who are also working on engineering retro elements. There are even newer systems such as bridge RNAs from Patrick Schu's lab at the Arc Institute and others. And so the field is taking on a multi-pronged approach to try to solve this problem of how to efficiently introduce gene-sized cassettes at single sites that are defined in advance into the human genome in living cells. So our initial salvo into this, again in collaboration with Wen Shui's lab, was template jump prime editing. And this is something that adopted a strategy seen naturally with non-LTR retrotransposons, which use a nicking site that then primes reverse transcription of the retro element transcript. And that retro element transcript can then terminate in the reverse complement of a second primer binding site at a second nicking site that can then reverse prime second strand synthesis. And so we generated, tested, and implemented a similar system using a PEG RNA that also encodes the reverse complement from a second nicking site, which is already built into the PE3 editing system. And that flap, single-stranded nick on the originally non-edited strand is complementary and can again reverse transcribe. Since this was published in 2023, I'm not going through it, but we were able to get insertions at 500 base pairs, 800 base pairs, not multi-kilobase with any great efficiency, but nonetheless higher efficiency at the lengths of, for instance, individual exons. So even though it can help in many contexts, it's very site dependent, it's variable in its performance. It is sometimes accompanied again by indel byproducts. And so even though we were excited at the utility of this, it did not solve every issue associated with the challenge of introducing large insertions or overwrites. So now I'm going to switch to an unpublished story that we have on a different approach towards large insertions. And it begins with the basic question of when you have paired prime edits, and there are many reports previously from multiple groups showing that you can have paired prime edits where the flaps are pointed inward and introduce edits in various configurations to the sequence that lies in between. But then we asked whether if we introduce flaps that are different from the native genomic locus and then build complementarity into a DNA donor to those flaps, do we actually drive any editing events such as insertion of what lies in between those A and B sequences that are cognate to the flaps that prime editing installed? This is again work that was spearheaded by Bin Liu, who's a jointly advised postdoc between my lab and Wen Shui's lab. And so as you can see, this has been an ongoing and very productive and rewarding collaboration between our groups. And so the initial attempt not only worked, but it worked surprisingly well. And so this is just an agarose gel analysis where we do PCR across the edited locus. Unedited DNA is shown at the bottom, and the insertion sequence is shown by this band, which is marked in red. And this is where we have PCR donor, double-stranded DNA, with cognate ends, cognate to the flaps that were installed by prime editing. When we use a plasmid DNA donor, it no longer supports this edit, indicating that the free ends are in fact important for the insertion event. When we include nickase Cas9 that does not have a reverse transcriptase fused to it, we do not get any product. If we use a PEG RNA that generates flaps that are not cognate to the DNA donor so that A and B no longer match with the flaps that were generated, we again lose the insertion. And this is just in HEC293 cells at the AAVS1 locus, suggesting that this is working. This is not a quantitative analysis because PCR products of different lengths can be amplified with different efficiencies. And so we then turned to droplet digital PCR to quantify editing efficiency. And we get an average of roughly 50% editing efficiency in this particular site and insertion event. We did junction PCR and Sanger sequencing across the junctions and showed that the editing events were precise and as predicted. And then we tested donors that became increasingly lengthy, going from 0.8 to 2.2 to 4 kb, and we retained substantial efficiency in these edited events, which we again analyzed in comparable ways, showing that the expected insertions were in fact happening. And so if we switch to a different cell type and a different locus, for instance, this is a T cell line known as JERCAT, and we're introducing the same 0.8 kb donor into the T cell receptor locus, we can once again observe editing as expected and as observed previously in HEC293 T cells. There are a battery of small molecules that have been reported previously to affect molecules that are involved in various aspects of DNA repair and other aspects of DNA metabolism. One of them is an inhibitor of DNA PK, known as EZD7648. And DNA PK is a non-homologous end joining factor. And non-homologous end joining could be potentially an inhibitory competing pathway with the types of repair elements that we are trying to detect. And so Bin tested the effect of AZD7648 on insertion frequency and saw a dramatic increase, which quantitatively, based on ddPCR and multiple replicates, averaged about a doubling of the editing efficiency, again, at this particular site for this particular edit. We are aware of the fact that inhibiting non-homologous end joining can have detrimental effects, and so we flag this not necessarily as a path to in vivo applications, but more as a mechanistic insight into potential ways to promote and optimize the efficiency of this type of insertion editing. A wider screen of multiple compounds is shown here. This is again the same 0.8 kb insertion. And so no donor, we get nothing. No inhibitor, we see the same insertion that we had seen before. And then various inhibitors with the concentrations shown, as well as the identity of the endogenous protein that is inhibited by that particular compound, is shown here. And so I just want to once again flag AZD7648 as being a reproducible enhancer of this type of insertional editing. But I also want to flag in particular two others, namely nocotazole, which is a microtubule inhibitor that disrupts progression through M phase and stops the cells from cycling. This is potentially useful because under non-cycling conditions, cells in general are poor to non-existent in their ability to support homology-directed repair. In addition, BO2 is an inhibitor of RAD51, which is a crucial player in homologous recombination. And editing is observed in both of these, under both of these inhibitory conditions, suggesting that the mechanism that is involved is independent of homology-directed repair, although deeper analyses will be required to understand this fully. What we had shown you before were primary products of PCR, that is, they have blunt or nearly blunt DNA ends. To ask whether single-stranded DNA ends can promote this, we used synthetic donors that incorporate phosphorothioate linkages partway through, and then used lambda exonuclease to chew away until it reaches the phosphorothioates, which block the exonuclease's progression, leaving single-stranded ends that are complementary to the flaps that were generated. And this worked, but the overall efficiencies that we saw were not enhanced relative to what we had seen previously with completely double-stranded donors, indicating that single-stranded ends are certainly compatible with, but they do not appear to be essential for the insertion of the donor in this context. So another question we wanted to ask is that if overlap between the flaps and the ends can promote a single donor to be built in, can we increase the potential length limits of this by using overlapping donors, that is two donors that themselves have overlapping sequences in the middle, so that you can put a double donor together and get insertion into these cells. And again, in a AZD inhibitor promoted fashion, the answer was yes, the double donor does drive the efficient insertion of, in this case, again, a 0.8 kb insert into the AAVS1 locus. This is droplet digital PCR quantitation. And then because it's we can look at the junction reads by Illumina sequencing and get, of the junction reads, we get most of them being precise insertion. We get some degree of indels, as with most prime editing based technologies, but they are again relatively low and comparable to other types of prime editing based platforms. We then tried a triple donor. In this case, it's an alpha-1 antitrypsin insert with an iris in front of it, total size 1.9 kb, put together as 3 different donors with the outer sites complementary to the flaps again, and then carrying similar 30 base pair overlaps within the donors themselves. We tested it as a single donor and as a triple donor, and both of them again worked in an EZD inhibitor promoted fashion. And so basically what this leaves us with is an in vivo editing platform that we can consider to be at least somewhat analogous to a process that many in the audience have undoubtedly used for plasmid construction, namely Gibson assembly, which uses overlapping sequences and polymerases of various kinds to put together an insert in a cloned plasmid. And so what we have here is essentially a mashup between prime editing and Gibson assembly. And so, we can consider the name Gibson editing or the name prime assembly. And we are coalescing around the terminology for this as prime assembly. And I just want to note that independently, Sebastian Levesque and Dan Bauer at Boston Children's Hospital have done very comparable work that they presented publicly at ASGCT a few weeks ago. And so we are grateful to them for communicating their results ahead of publication. And they have a very overlapping, similar, and complementary story to the one I'm telling you today. So when we go into next generation sequencing analyses of the editing events, we can do this in a number of ways. What's shown here is nanopore sequencing for an 800 base pair insertion using different categories of donors, double-stranded DNA without any phosphorothioates, the overhanging dsDNA with phosphorothioates, again generated by lambda exonuclease, as shown here. The same double-stranded DNA donor with phosphorothioate linkages, but not treated with exonuclease, and then the donor involving 2 overlapping donors, so the double prime assembly type of approach. And this is the amplicons associated with this were subjected to nanopore sequencing. And as you can see, product purity is pretty good for the standard double-stranded donor. Whenever we involve phosphorothioates, either with overhanging or non-overhanging ends, we start seeing some additional byproducts at the site of either priming or phosphorothioate incorporation. And then some additional byproducts appear whenever we have the double donor. And then when we add these up and compare indels to precise insertions, we do get indel byproducts, but they are again comparable to those seen with many other types of prime editing based editing platforms. If we do Illumina sequencing only across the junctions, we cannot Illumina sequence the entire insert because it's too long. But if we do the across the junction at both flap A and flap B, we can again quantitate those that are accompanied by either precise editing and expected junctions, as opposed to those that lead to indels. And as you can see, we do get some indels, but they are relatively modest. And this is a sequencing of a G-block control, a synthetic DNA, which is estimated to have an error frequency of only 1 in 5,000, indicating that many of these apparent indels likely come from sequencing errors, not true indels in the template themselves, but that will require additional analyses. And it's similar whenever we use the the single-stranded overhanging ends in our donor template. We can also ask, do we get functional protein from an edited locus? And to do this, we inserted GFP followed by a P2A self-cleaving peptide in frame into exon 2 of the ACT-B locus that encodes an isoform of actin. and just simply ask, do we get GFP expression? And the answer is that we do, and it is enhanced by the DNA PK inhibitor, and it is also dependent upon the donor, or sorry, the prime editing that accompanies the donor delivery. There is a lot more that we can do in terms of donor optimization. That includes using synthetic single-stranded DNA donors, in this case, 60 nucleotides with 25 base pairs of overlap, and 35 nucleotide overhanging ends. And so the overall insertion is 95 base pairs. The PEG RNAs are 90 base pairs apart, so you accompany a 90 base pair deletion with a 95 base pair insertion, which is effectively a sequence overwrite. And so we cannot distinguish between unedited and successfully edited by PCR because they're too similar in size. But nonetheless, we built in a high specificity restriction endonuclease site into the overlapping sequence, ISCE1 from yeast. And initially, we can take the PCR amplicon and subject it to ISCE1 digestion and show that we get the expected digestion products as a predominant product, indicating that editing occurred very efficiently. And we are in the process of confirming this by direct sequence analysis. So if we really try to push the length limits, we can go to something like the dystrophin gene, which is one of the longest cDNAs in the human genome at greater than 11 kb. It actually pushes the limit of efficient PCR amplification. But we made a dystrophin donor carrying flap homology and tried to insert it into the AAVS1 locus, again in HEC293 T cells. We cannot do, well, it's easiest to do junction PCR, and we can detect successful edits at both ends. We can sequence those amplicons across the junction and show that they occur accurately. And we can again show that in an AZD promoted fashion, we get insertion by long-range PCR, which is not a quantitative analysis, but nonetheless gives rise to the expected product. And then last but not least, we can insert a chimeric antigen receptor into the track locus in HEC293 T cells. So I'm just going to stop here in the interest of time and summarize. Prime assembly is a new approach for multi-kilobase insertions, and now there's lots to do and many questions to answer, including mechanism, what is host factor involvement, what's the polymerase that's accomplishing this. There's a lot to do to optimize it and engineer donors, test its efficacy in primary cells and eventually in vivo. DNA donor toxicity will likely be a concern as we try to go to more challenging contexts, and that will have to be addressed, and many others. And with that, I'd like to thank members of the lab, especially Ben, who did this work and is about to start his own lab at Ohio State in just a few weeks. It was other members of the lab made important contributions, as well as did some recent alumni from the lab who are now pursuing their careers elsewhere. This was a great collaboration with Wen Shui, and it was contributed to by other members of Wen's lab, including Andrew Petty and Sheena Zhao. We are grateful to Sebastian Levesque and Dan Bauer for communicating their results on a similar system. We got a lot of help in various ways from the Watson Wolf Labs at UMass, and these people for funding, and you for your attention, and I will take questions.
Thank you very much for that wonderful talk, Dr. Sontheimer. I guess my first question is, what are the obstacles limiting insert size, and is there a linear relationship between size and integration efficiency, or is there some sort of threshold where it drops considerably?
It's very, very context dependent. You can't really provide single answers to these things, especially because they will differ somewhat between HDR, transposons, integrases, and so on and so forth. In some cases, for instance, with transposons, length limitations might be imposed by the transposition machinery itself. HDR, there's a number of published reports. It does appear to get more difficult as you get bigger and bigger, but the relationships are not clean and linear. It's very, very context dependent. From the standpoint of prime editing, we don't really know necessarily what the biggest barriers are. You can potentially make a very long flap, and you could potentially do that with high efficiency, but since you're relying so much on the cellular repair machinery to process that flap and drive its invasion and subsequent resolution, it could be that the bottleneck is in the DNA repair machinery rather than in flap synthesis. And the field is actively pursuing questions such as this.
Thank you very much. So for prime assembly, how far can you take it?
Yeah, I mean, we don't know yet. 11 kb is farther than we really thought would be possible, and we haven't pushed it longer. So we simply do not know.
Have you noticed anything that could be a potential obstacle if you were to take it further? Or again, is that very situational?
Well, a big obstacle is going to be the requirement to co-deliver a DNA donor. DNA delivery in primary cells and in vivo is a major challenge for a number of reasons, but very much including the fact that cytosolic DNA is highly toxic to cells. It is sensed by innate immune activity. This has been an issue with homology-directed repair as well, including in CD34 positive HSPCs and T cells and other contexts such as that. And so the requirement for a DNA donor is going to be a substantial one. The fact that single-stranded donors can work to some extent may ameliorate that to a considerable extent, but that will certainly be a concern that we will have to address very stringently and very rigorously.
Thank you very much, Dr. Sondheimer, for that wonderful presentation, for answering the questions, and for being here with us today. In the interest of time, I believe we will have to wrap it up here. I'd like to take this opportunity to once again thank our symposium sponsors, GenScript and BD Biosciences, as well as our panel of speakers, Benjamin Kleinsteiver, Mitchell O'Connell, Daksha Patel, Carrie Eckert, and Eric Sondheimer for sharing all of their wonderful insights. I want to thank all of you in closing who joined us today and participated in our discussion. Remember, the full recording for this event will be available on the scientist's website, and we will email you the link for the on-demand version when it becomes available. Please also make sure to follow the scientist on social media to stay up to date on future webinars and other events. Thank you, everyone, and have a pleasant day.